Good morning. Morning. How are you all doing today? Hopefully you guys had a weekend. What I mean by that, mostly fun. Not a crappy weekend. That's what I'm hoping for. Hopefully you guys did not have a crappy weekend. Yeah, I never really think of texting y'all as work. I mean, yeah, you guys can text me as much as you want. And the fact of the matter is so far this class has been very good about asking me questions, and I love it. Like, that makes me happy, super happy. It's a weird oddity of mine. I want students to contact me. I just remember when I was a student and I was taking phys, and I'll give you guys just a little bit of these stories of physiology, because physiology is freaking hard. It's hard. It's still hard for me in some aspects, right? When I took it, we had our lab groups, but I had a small study group to the side. There were a bunch of students that weren't part of my lab group. And, there was basically 5 of us that would get together about 3 times a week. And we had a big whiteboard up on a stand. And we'd always meet up, always, no matter what was going on with everything else. And we had one guy in the group that brought food every single time. We had another guy in the group. His name was Hero. He was our hero. He brought us beer every single time, and we studied. We drank a beer or two, and all we did was just did increased decreased no change questions for hours upon hours. And yeah, we had a lot of fun doing it, because when you're studying, you should kind of, work your patella off. Never do it alone, and that's kind of where this all goes. Is text me, because you should never try to study physiology by yourself. If you start going in a circle, right? You're studying something, you don't understand it, you're trying to make sense of it, and it's just not making sense. You just get more and more frustrated, and then the next thing you know, you're so frustrated you can't study anymore. Anybody ever been like that? You can't even go to a different subject. And it's just like, we've all been there. It's part of being a student. And so instead of getting to that point, just text me, right? I had a number of students that were like, I don't understand this homework question at all. And there you go. I'm going to try to break it down. I won't necessarily give you the answer because it's a homework question, it's graded, but I'm sure I'm going to lead you to that answer by saying, here's what we did in class. Remember this part. What do you remember about this? So forth, so on. So, yeah, I mean, I do hope you guys start enjoying your groups and appreciating each other in your groups and using each other and leaning on each other. Um, the guy that always brought food to my physiology study groups, Brad Kramer, well, let's just put it this way. He was my best man at my wedding, and I was his best man at his wedding. So, I mean, we spent a lot of time together. And I think that's how physiology should be, because it's hard. So speaking of, let's do a little bit of review, because I always like to do. Hero, oddly enough, no, Hero, I don't even know where Hero is to this day. So he was a super cool cat. Being a hero somewhere, you got it. He was a good guy. We did not stay in contact after that class was over. It happens. All right. So what we want to remind ourselves is a approach we learned last week that helps explain a lot of the endocrine system. And that flow chart goes as such. Everything in our body, everything starts as with a stimulus. Nothing can happen in our body without a stimulus. Everything in our body starts with a stimulus, period, end of story. And in order for our body to be able to recognize this stimulus, this change of event, whatever it might be, what does our body have to have in order to detect these change in stimuli? Help me help you. You guys answer that. You got it. We got receptors for the stimulus. Now, where are the receptors for these stimuli? It just depends on the endocrine loop. Now, a large majority of what you guys learned about last week had to do with that hypothalamo anterior pituitary axis. And so in that scenario, the stimulus will always communicate with the hypothalamus, always. So the hypothalamus was that first integration center that we talked about, hypothalamus. Right, this is the first integration center that we talked about. So it's going to take that information and decide what it needs to do with it. And what it's going to do with this particular stimulus is it's going to end up secreting a hormone into that primary plexus, into that little portal system, that little bloodstream that's going to now go down to the anterior pituitary. Travels just pituitary travels just a couple of centimeters, right? Doesn't go far, but there are receptors in the anterior pituitary for that hormone. And then the anterior pituitary is going to respond. It's going to be another integration center. Just kind of thinking of one of the homework questions people had questions about. This is going to be another integration center. So it's going to integrate this information and it's going to determine a response. Well, its response is pretty set in stone. If the stimulus is there from the hypothalamus, then it will respond by secreting that particular hormone. And that hormone goes into the bloodstream, goes out to the entire body. Pretty powerful, pretty magical. And it's only going to interact with specific areas because what makes a target cell a target cell? Again, it's the presence of receptors. So wherever we have receptors in the body, we call these target cells. And then these target cells are going to be the end of the road. There's no integration here. The target cells are basically just doing what they're told, right? So these now are going to be the effectors. If you're thinking about those normal physiological reflex loops, these would be considered effectors, and they are going to create a compensatory response. They're going to create a response. But why is it a compensatory response? Because its response is always going to go back and communicate. Why did I go to the stimulus? I don't know. Don't look at that. Don't look at that. Don't look at that. That didn't happen. I thought I was going to here. They're going to go back and communicate with these original integration centers. These are going to integrate this information and decide whether to continue the response or to turn it off. This is a negative feedback loop. Does that make sense to everybody? And again, this is just a review. I like to review. I love reviewing. I think redundancy is the key to learning. So we're going to do it pretty much every single lecture. Yes, they are always a negative feedback loop, except for oxytocin. Well, and except for GnRH, but that's only during a very specific time of the female estrus cycle. So we'll end up, we'll talk 99.9% about negative feedback loops. All right, so the very first hormone that we're going to be discussing about today, we're going to go ahead and just talk about the hormone, and I want you guys to do your best to remember and fill in this area of this template, this flow chart. But we're going to be talking about growth hormone. So now growth hormone is a little bit different in its integration, not completely different, because a lot of target cells are going to be secreting another hormone for things like cortisol and stuff like that. But growth hormone, it doesn't trigger the release of another hormone. It's going to cause the target cells to have direct responses. So we're going to go ahead and start with what hormone is going to be released from the hypothalamus. We'll talk about the stimulus in just a second. What hormone is going to be released from the hypothalamus that will end up stimulating the release of growth hormone? Who remembers this from last week? Short-term memory stuff. Yes, yes, so good, so good. So this is growth hormone releasing hormone. Growth hormone releasing hormone. And that growth hormone releasing hormone, there will be stimuli that cause it to be released. Again, we'll talk about those in just a second. We just want to go with the main loop. That's going to get down into that portal system. There are receptors for growth hormone releasing hormone in the anterior pituitary. And as GHRH increases, the anterior pituitary will release growth hormone. Anybody remember from last week, what are the target cells for growth hormone? Yeah, every cell in the human body has growth hormone receptors because every cell in the human body grows. I'm not talking about growing in mass. I'm talking about cell division. Mitosis requires growth hormone, and every cell in the human body will divide. So just that process of cellular division, cellular proliferation, cellular differentiation, all of these cellular, growth, replication, a lot of these are triggered by growth hormones. Pretty powerful, huh? In fact, let's just do something that's a little creepy, not creepy, I don't know. That's not the proper terminology, but trying to get you guys on the internet here. Let's look at somebody that is taking GHRH. So GHRH can be injected, and it's the new steroid. It's the new testosterone. And I want to see if there are any images of people. I don't know, people. Give me pictures of people. I want to see a person. Injecting himself with mushrooms. What's going on there? And actually, they normally go growth hormone releasing hormone. But yeah, so growth hormone releasing hormone. Now, testosterone makes those guys like big and huge and bulky kind of thing. This is a little bit along that line. But growth hormone releasing hormone makes you very trim, makes you really skinny, but also very toned and muscular. So you end up, a little bit more like this. A lot of people are taking GHRH. It is not legal, but they do it anyways. Yeah, there are definitely diets out there. There's a lot of under the table stuff with growth hormone. So anyways, just kind of a nice way to be able to think about its responses on target cells, all cells. And the primary response I want you guys to think about, we'll write a few more responses down here. Professional headshot, that's funny. The primary response that you guys will see with growth hormone down here has to do with metabolism. Okay, so the way I like you guys to organize your thoughts on all of these hormones released from the anterior pituitary is that they all relate to metabolism. In fact, most of the hormones that we end up talking about are relating back to metabolism somehow. In growth hormone, I mentioned this last week, do we want more glucose available or less glucose available? Good, right? Because if we want to grow, we need more glucose for that function. True story? Because glucose is how we make ATP. And actually, I haven't written that down for y'all yet, come to think of it, just as a reminder. This is a formula that should be a part of your biology background, but I assume all the time that students know this, and then I find out they don't all know it. So let's go ahead and just recover our memory. Our last memory on this is that we need to make ATP. And the simplest form of looking at how we make ATP is by looking at this formula called cellular respiration. And we're going to use this formula for cellular respiration, I don't know, about 1,000 times this semester. Cellular respiration is all about the process of making ATP. It's also known as aerobic oxidation. Aerobic oxidation. So it has to do with oxygen. So for cellular respiration to occur, everybody take a deep breath. Oxygen into the body. Why do we need oxygen? Because oxygen is required to make ATP. That's what's going on. So when we start it all off, take a deep breath in. Because when you don't remember something, what should you do? Just breathe for a minute. Sometimes it'll help you actually come up with the answer. Oxygen. Now, why do we eat? We eat for glucose. There are a few other things that we want to get out of the food, but glucose, C6H12O6. Oxygen plus glucose, we have some phosphates that are running around in our body. We've already made them into ATP. And there's some inorganic phosphates that are running around as well. This is basically just an ion, an inorganic phosphate. We store all of these inorganic phosphates in our bones. So we got a big storage of this inside the body already. This goes into a reversible chemical reaction, and the byproduct is ATP. We end up making like 36 ATP per cycle. This is all the Krebs cycle, crazy stuff, right? What's the metabolic waste? Because all chemical reactions create waste. What is the waste of this cellular respiration? Anybody remember? Yeah, lovely. It's carbon dioxide. And oddly enough, there is another waste product. We just don't think of it as waste typically, but it's a waste product in this chemical reaction. Not phosphate. The phosphates are all being used up. ADP, D stands for di, T stands for tri. So we start with 2, add 1, we get our 3. Yeah, the last metabolic waste from this is water. You guys are going to find out later when we get to the respiratory system how the CO2 and the water, how they're going to end up interacting in the body, and they end up making acid. So not always a good thing. But you'll notice respiratory system, digestive system, skeletal system, we got ourselves ATP, we got a bunch of CO2, respiratory system, and renal system. All of these systems are used to control cellular respiration and making sure that we get enough ATP. So here we are in an endocrine system format, and I want you guys to see that if I want to increase metabolism, I need more ATP for that function. So if I want more ATP, what do I need to do to glucose availability? Increase, decrease, no change. Yeah, does this make more sense for anybody that might have been confused about the whole loop between ATP and glucose? Is this clear? I need more glucose to make more ATP. And then what about oxygen? What's going to happen to oxygen demand? Increase, decrease, no change. Lovely. So as I'm burning through ATP, I need more oxygen. You might see that they might actually, if they're hyper metabolism, right? If they're going crazy with metabolism, which you see in thyroid patients, for instance, hyperthyroid patients, they're going to have deep, fast breathing associated with that. One more thing to think about is that ATP is energy. And as we use energy, all energy is consumed to make work. So all energy is consumed to make work. This is a physics thing, right? When we burn energy, we're using that energy for work. And this is through the entire universe. This is not just in your body. Gasoline, energy, we burn it, we get work. But what's a byproduct? Not all energy can be used for work. There's actually a lot of energy that's wasted. Okay, gas energy is very inefficient. There's always a lot of waste. Coal energy is inefficient. There's always a lot of waste. ATP is a little bit more efficient, but there's still a lot of waste. And what is the waste product of burning energy? Always. Lovely. Good job, Parker. It's always heat. No worries, Tariq, man. Carbon monoxide is, from the breakdown of fossil fuels, but your body doesn't make carbon monoxide, thank goodness. All energy consumption will always make heat. Heat is wasted energy. Thank you, sun, for all of your wasted energy, because the sun is sending us a lot of heat through those UV rays, which is part of a wasted energy source. Just think about it this way, right? When you exercise, what happens to your body, your core body temperature? Increase, decrease, no change. We all know this. You're going through your energy, therefore you produce heat. What about when you get too cold and you start to shiver? When you shiver, that's a muscle contracting, relaxing, contracting, relaxing, contracting, relaxing, contracting, relaxing over and over and over again. It's your body's response to try to increase the amount of available heat in your body because those muscles are just working. All right, does this make sense? This will come back to play a role in our endocrine system stuff. So this is not far-fetched here. All right, cool. So GH, what we want to do is we want to somehow increase glucose availability. There's a lot of words that we learned that could do this. Increase glucose availability. Cool. There will be more things, but what sort of words did we learn allow us to increase glucose availability? Let's get back to our lecture so we can be done with this part. But I want you guys to see, right? Direct effects at the adipose level. Man, I could talk, I swear. At the adipose is fat tissue. Do we want to store more fat or do we want to break down more fat to release potential glucose? Do we want lysis or genesis? L or G? That's awesome. Great job. Muscles. Let's drop down to liver. I think I said I was going to come back to muscles last week and I never did either, but I will this time. In the liver, remember the liver is the #1 storage site for glucose in the body. It's not stored as glucose per se, it's stored as glycogen, but glycogen is just big long chains of glucose over and over and over. It's the potato of the human body. Okay, potatoes are they're made of starch, which is glucose. Big long chains of glucose. So yeah, the liver is the potato of the human body. I love that. So glucose, we want to break down glycogen to get more glucose. We want to use the liver to take things that were not glucose, like amino acids, like glycerol from fat, and we're going to make those chemical products into glucose. Gluconeogenesis, boom. We do those things, we make more energy available. All right, now, why would we want to decrease glucose uptake into muscles? I want you guys to think about this. Why would that be a good thing? Yeah, you got it. Perfect, Andrea. Love it. Andy, that's great. We want to make glucose available elsewhere. Well, what is the #1 consumer of glucose in the human body? Does anybody know this? What organ consumes more glucose than anywhere else? Yes, the brain is going to consume over 50% of your glucose in any given time. And as you are growing and developing, the brain needs even more glucose. So we decrease the amount of glucose going to our muscles so that there's more glucose available for the brain. Yes, no, maybe so? Does that make sense to everybody? It doesn't mean muscles aren't using the glucose. They are. Muscles store glucose away as glycogen as well. So muscles are pretty lucky in that regard. So if we get more amino acids in there, then we can actually use the stored glycogen and those amino acids to make more muscle. So growth hormone will be released just by exercising in order to make more muscle. So they're pretty cool that way. All right, so there are some indirect effects. I mentioned this one last week, is that when growth hormone is released, it will always cause the release of insulin growth factor, insulin-like growth factor to be more specific, IGF, IGF-1 to be even more specific, and then IGF-1 is essential for all of these following processes. In the presence of growth hormone, IGF-1 will also cause cell division, cellular differentiation, causes more proteins to be made, causes bones to grow, stimulates muscle to grow, cartilage to grow, fat tissue to grow. Whew. There's a lot of potential growth because of the available energy. All right, so this is the one that I really, really like in terms of the total package. This one also talks about a lot of the stimuli for growth hormone release. So let's talk about this. Variables that influence the release of GHRH from the hypothalamus. The first one on here is age. All right, still makes you think. What does it mean by age? Who's growing the most? An adult or a kid? Yeah, adolescents, kids, right? So whose growth hormone is going to be higher? A kid or, yeah, so does that make sense? You guys see the logic there? Kids have extremely high growth hormone release. They're growing. Their brains are developing. Their muscles are developing. Their bones are developing. Their cartilage is developing. Adults still have growth hormone. Don't think we stop producing it, but we're not growing more in height. We still have to develop, and our brains are constantly growing and developing, so we still have growth hormone. So don't think we stop having growth hormone when we hit puberty. Time of day, remember what I mentioned about this being on a circadian rhythm, and that most growth hormone is released while you're sleeping at night. Nutrient levels in the blood. All right, so this has to do with amino acid levels and glucose availability. All right, now there's a homework question that's related to this. So please go in, I need you guys to be thinking about these nutrient levels and their availability and how it would work in terms of making sure that more growth could occur. Stress and exercise. We've kind of already mentioned this, but if you are exercising, which is a form of stress, by the way, or any other sorts of stress, oddly enough, as you exercise, what do we want to do to the release of growth hormone? Why do we exercise often? Do we want to store away more fat? Is that why we're exercising? Or do we want to break it down? Break it down. Break it down. Right? So does that help you understand that if you're stressed out, most of the times, stress is actually well, we'll talk more about stress when we talk about cortisol. But let me tell you this: the number one stressor on the human body, pretty much when it comes down to stress always causes hypoglycemia. Okay, the number one stressor on the human body is hypoglycemia. Hypo meaning low, glycemic meaning glucose, emia meaning of the blood. So hypo is low, the glycemic is glucose, and then the emia, the EMIA, means of the blood or in the blood. So this means low blood glucose. That's the number one stress on the human body. And if you are hypoglycemic, what's the body going to try to do? Increase blood glucose levels or decrease them further so you die faster? Does that make sense? So hypoglycemia and stress, we want to release growth hormone to make it so that we have more glucose available. Yes, no, maybe so? Awesome. Now, when we look down here, right, increased glucose availability, that's this side of this flow chart. So increased lipolysis, increased glycogenolysis, increased gluconeogenesis, this is all about making sure there's more blood glucose available. So when you go here and it just says glucose availability is increased, make sure as you guys study that you understand that has to do with each one of those: lipolysis, gluconeogenesis, glycogenolysis. Yes, no, maybe so. Just because we didn't write it down doesn't make it true. We're just summarizing to make this easier to see. You guys with me on that, please? Cool. And then when you go in to fill in your stimuli, try to do this all by memory. Try not just to copy everything. But now we're looking at things like age-related, time of day related, right? Exercise and stress. This is one of those weird words. There we go. Stress. So there are a number of different stimuli that you guys can learn about. And there's even more stimuli than this that you can learn about online. Nutrients. So like, is glucose available? Are amino acids available? I mean, geez, if amino acids are available, let's use those to make something. Let's not just let that go to waste. Let's use it. Oh, very cool. All right. That's growth hormone, always a table. Boom. Okay. So let's move on to the next one. And I want you guys to see right off the bat, what is this? What is this distinct pink bubble gum? What did we learn last week? Yeah, perfect. This is colloid. And if we find colloid, what is the full circle called right here? What is the full circle? The cells and the colloid. It's a follicle. Good. So, Tariq, you're not wrong in terms of what these outer cells are called. These were follicular cells. The full circle is a follicle, and the follicle is made up of follicular cells and colloid. Now, for the really hard word from last week, what is colloid storing? What is stored inside of colloid? Anybody remember that word? It is a protein. Lovely. What is that protein's name? It's thyroglobulin. Okay, for anybody that is just over there going, I don't know, Dr. H. Dude, you're fine. You're fine. That's okay. Take a deep breath. I'm not trying to, I'm not trying to make you guys feel bad. I'm always just trying to throw out those key words that if you don't remember, that's a good check mark for I need to study this some more. Is everybody clear on that? You don't have to know every answer to every question that I'm putting out there. I'm just trying to make sure that you guys kind of have this. Okay. Study guide. Thyroglobulin. You will be tested on thyroglobulin. We're going to go into a lot more detail about what thyroglobulin is, but it's stored thyroid hormone. There's a reason for that. There's a reason for that. In fact, let's go ahead and just kind of quickly describe the reason why it has to be stored the way that it's stored. And that reason comes down to the classification of this hormone, T3, T4. This is thyroid hormone. T3 stands for tri for the 3, iodo. What does iodo tell people? What chemical or molecule would iodo have to do with? It's iodine, which is why we have iodinized salt. So iodine comes from the food we eat, and it helps us make our thyroid hormone. Without it, there is no thyroid hormone. And then thymine is the protein that is going to be kind of broken down. Tetra tells us 4 iodo. So that's T4. So I have a T3 and a T4. We'll learn more about the fact that, there's only one of these that is active. The other one's in inactive form, blah, blah, blah. We can also abbreviate this as thyroxine. So that might be a word that you'll see on tests or homework. But what I'm going to try to remind you guys of from last week is what classification of hormone is this? As a hint, what I'll tell you, this is actually not a steroid. Great try though. But thyrionine is a modified amino acid. So what does that tell us? It's a biogenic amine. Good. So this is a biogenic amine. What did you learn? Most biogenic amines are polar or nonpolar. Polar. And what was the one exception to the rule? They're almost all polar. They're almost all polar, but what was the one exception to the rule? Thyroid hormone. You guys are doing so good on your memory. I love it. 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 So everybody, let's just summarize this. Thyroid hormone is a biogenic amine that is non-polar. It is very unique in that aspect. Most amino acids are extremely polar, but not thyroid hormone as a biogenic amine. It is non-polar due to the presence of those iodine and due to the type of amino acid it is made from. So what do we know about non-polar and water? Do they mix? Yeah, they don't mix. What else do we know about non-polar stuff? Can non-polar hormones, yes, can non-polar molecules simply pass through cell membranes? Yes or no? Sure they can. Thyroid hormone, if it was not stored, is non-polar and it would just go out into the bloodstream as it was made. Does that make sense? It doesn't need thyroglobulin in order to travel, Heather. One step at a time. There's great thought process, but that's not what we need thyroglobulin for. We actually have a different protein in order to transport it around. So keep with me, everybody. If we didn't store it away as we made T3, T4, it would just simply go from an area of high to low. So as we made it, it would be diffusing into your bloodstream all the time. But thyroid hormone is one that we want to release only when we need it. Only when we need it. We need to regulate when thyroid hormone is released, but how the hell can we do that with a non-polar molecule? It's really hard. Well, what we do is exactly what Heather just wrote. What we do is just like in the bloodstream, in order to transport a non-polar molecule around, we need a carrier protein. Well, what if we want to store it away? Well, let's store it away on a storing protein. Does that make sense? And as long as it's bound to that protein, can it get through the cell membrane? No, it's bound to a protein. So it's stuck there. And that's what thyroglobulin is. That's why we get these big, huge, just vacuoles filled with protein, thyroglobulin. And as the T3 and T4 gets made, it's going to bind to that thyroglobulin, and it's going to be stored here until we need it. Pretty cool, huh? Pretty powerful. Flowchart. From the hypothalamus, what hormone will be stimulated when we want more thyroid hormone? We'll talk about the stimuli and the responses more in detail in just a second. What do we got going on here? What hormone? Well, we do know this is hypothalamus, so it's going to probably be an RH. So we'll go ahead and write an RH in here. Yeah, this is TRH. Does anybody remember what TRH stands for? What does the T stand for? good. It's actually not thyroid releasing hormone. I like that you went there. It's basically thyroid releasing hormone, but it's actually thyrotropin, which is important to take a note of because thyrotropin will end up telling you that it's a hormone that causes a release of another hormone, and it's going to end up going through a bunch of different cycles. Yeah, exactly. So that TRH is now going to travel just a couple of centimeters through that primary plexus, that portal system, down to the anterior pituitary. And in the anterior pituitary, we're going to end up releasing what now? What hormone? TSH. Yes. Which gets into the bloodstream and goes to, where are the target cells for TSH? This is a little bit more complicated. We did cover it last week, but we covered it in the lab. I'm just seeing if anybody remembers. What gland would be the target for TSH? Let's go there. Yeah, thyroid gland, thyroid gland. And does anybody remember what part of the thyroid gland makes T3, T4? It's right here on this slide. What cells of the thyroid gland make T3, T4? Good. So where will the target cells be for TSH? They're the follicular cells. Does that make sense? All right, so we write that into our flow chart. Follicular cells of thyroid gland, and thyroid hormone will be released. T3, T4, and you can notice that there's, an extra arrow in all this. T3, T4. Oh, by the way, growth hormone, releasing hormone, we forgot to put in. We have, just depending on growth, increased glucose availability and growth. Well, we're going to have negative feedback loops for all of this. But back over here, T3, T4, that's where we get our negative feedback loops, is from if T3, T4 is too high, TRH, what's going to happen to TRH? As we elevate T3, T4, the body's always going to try to revolve it around set point. Beautiful. And then what happens if T3, T4 gets too low? What's going to happen to TRH? It's going to increase. So we'll come back to that. That's going to be a very important part of the lesson plan. But T3 and T4, again, is all about energy availability. T3, T4 actually is a primary hormone that helps to cause more cellular respiration. What? More cellular respiration? What do we know about that? It is all about energy. So we're going to do everything we can to increase energy availability. And we're not just talking about glucose here. We're going to talk about a lot more of the energetics, energy availability. But hopefully you can see the similarities with growth hormone. So for instance, lipolysis, yes or no? Increase, decrease, no change, or yes or no, I don't care. What about lipogenesis? Yes or no? Good, good, decrease, I love it. What about gluconeogenesis? Increase, decrease, no change, the gluconeogenesis. You guys got the point, right? Energy availability, I love it. How do you guys feel about that? Like if I tell you that there's an energy availability that needs to happen, now you guys are starting to think about these cellular processes. Everybody feel okay? Or at least that you understand what I'm asking from you and that you need to study it some more? Yes, no, maybe so? You know where I'm trying to lead you, because the endocrine system is relatively disorganized. It's just a whole bunch of different hormones. So how can we find something that's similar to make it a little bit easier to study? That's what things like these flow charts and this idea of energy availability, that's where it comes into play. All right, we'll come back to this. Actually, let's do one more thing real quick. T3, T4. We said this is made from iodine. Yes? No? This is made from iodine. So what else is going to be required for this system to even work? I'm going to have another flow of information, and really it goes up here. So let's draw the arrow up here. Breaks. I'm going to have to consume iodine in my diet. So I'm going to literally write iodine consumption. If you do not consumption, I don't know, T-I-O-N? God, that sounds right. Sure. How do you spell the? Sometimes I wouldn't know. So what would happen to the production of T3, T4 if you decreased your iodine consumption? What would happen to T3, T4? Increase, decrease, no change. So let's do this in flowchart form. This is what makes this so powerful. I'm going to use a green, and I'm going to say we decrease our iodine consumption. And therefore, the follicular cells can't make T3, T4. So what's going to happen to that patient's T3, T4? Increase, decrease, no change, y'all. Let's get a little bit more responses from everybody. Does that make sense to everybody in the classroom? I'd like to have everybody at least just give me a little bit of, you know, telepathy. Telepathy? No, that's probably not the proper term. I don't know. What is it when you talk to somebody with your mind? Anyways, there you go. Just let me know. Now, what do we know about negative feedback loops? If this patient starts to drop their T3, T4 levels, what's the body going to be trying to do to TRH? Increase, decrease, no change. Yeah, man, this patient's TRH levels is going to be elevated. TRH, in turn, is going to be trying to increase, not to mention the negative feedback loop, but what's going to be going on with TSH? Increase, decrease, no change. So they're pumping out TRH, they're pumping out TSH. Will that cause more T3, T4 in this patient? Yes or no? The answer is no. The answer is no. Why? Because where's the root of the problem? The root of the problem is lack of iodine. So in the medical field, what are you going to do with this patient? Just a thought process, little logic. Yeah, just give them iodine and they'll be fixed. Pretty cool, huh? So they have drop T3, T4. It's called hypothyroidism. You could just give this patient iodine. How are you able to determine what was wrong with this patient? Is they do blood panels. This is what makes endocrinologists look so smart. They just do a blood panel. And what did they test for? They tested for the amount of T3, T4, TRH, and TSH. And if they see that there's a drop in T3, T4, there's elevated TRH, there's elevated TSH, there's really only two things that can cause that group of panel. One, loss of iodine consumption, and two, cancer. So they just do some further studies, some further tests, and hopefully they find that it has to do with iodine. Crazy, huh? Love it. Why is that? Traditional lab tests, so traditional lab tests will often just test for two of these often. You might get it in a roundabout way because there's other tests for TSH out there besides directly testing for TSH, Heather. Does that make sense? But the rest of that question, I don't have an answer to because I, never actually practiced endocrinology. So, why certain things are on certain blood panels from a scientific standpoint, it's because some things are very difficult to measure. Like to create these chemical assays for particular hormones, they degrade so fast, they're really hard to detect because it's just really hard to make an in-lab assay to detect it. So we ended up with a lot of blood panels that we use what we can. Does that make sense? Kind of. Okay, cool. We'll come back a little bit more to this flow chart here in a bit, but I hope you guys are feeling pretty comfortable with it. We're going to continue using it. What do we know? The hypothalamus is releasing thyrotropin releasing hormone, which now is transported just a couple of centimeters down to the anterior pituitary, which has receptors for TRH, causing it to release TSH into the bloodstream, which is now transported all the way down to the thyroid gland. But what specific part of the thyroid gland? That's a picture of it. The follicular cells of the thyroid gland, and those follicular cells are going to produce more T3, T4, which now go out to the target cells of the body. You're going to be going through the flow chart for the target cells of T3, T4 later. Right now, I'm just trying to get through the hormones, and we'll fill in stimuli and the rest of the responses. All right, so another hormone or another part of the thyroid gland that you guys were asked to identify in lab last week are the parafollicular cells. Para meaning surrounding. So parafollicular, these are cells that surround the follicle, not surround, not because the follicular cells are part of the follicle. We're not looking for the follicular cells. We're looking for cells that are outside of that even. So if you go up here, there are no parafollicular cells in this particular picture. There's no cells that are kind of lying in the world between. But look at this picture right here. See how we have this really beautiful follicle, colloid, and all this stuff. And then we got this cluster of tissue and cells in between the follicles. Yes, no, maybe so? These are called parafollicular cells. They're also known as C, as in the letter C. They're called C cells. It's actually what I learned them as many years ago, C cells, because they secrete the hormone calcitonin. And I'm not going to retest you on calcitonin. That should have been an AMP1 thing, but I will remind you guys that calcitonin is all about calcium. True story? Does that make sense? You guys remember that from AMP1? Just if not, there's your little reminder push. Calcitonin is all about calcium. And calcitonin, actually, let's what do I of that will help you? Let's just do. I glitched. So I just glitched real quick. So that means you guys probably couldn't see my screen for a second. I could hear it, a feedback in my ear. That's how I know. Is my screen available to you guys? All right, cool. So let's just do a quick feedback loop, and this will help you understand calcitonin a little better. Goodness gracious me, oh my. Okay, so the quick just feedback loops you guys learned about in AMP1 is that we have a stimulus that acts on a target, and then that target is going to respond. This is called a simple reflex, right? This is a simple endocrine reflex. The target in this case is both where we find the receptors and it's an integration center. Why did I go, I guess respond, whatever, response. So in this particular case, the stimulus is a change in blood calcium levels. So blood calcium. Blood calcium levels can either increase or they can decrease. Those are the two ways that blood calcium levels can change. Does that make sense? We could either have too much blood calcium or too little blood calcium. Of course, there's no normo as well. An increase in blood calcium is called hypercalcemia. Hyper meaning above. Lots of things could potentially cause hypercalcemia, but Heather, especially, it's cancer is one of the leading causes for hypercalcemia. Those cancer cells can cause, basically they, hijack the body and say, I need more calcium, so they end up causing constant breakdown of your bones, and it's no bueno. So hypercalcemia, if there's an increase in blood calcium, but like in normal, just everyday life, you can still have a little bit of hypercalcemia. Just it's going to start to elevate. So the target cells that are detecting hypercalcemia, those are the C cells. Okay, these are the parafollicular cells. And as they detect an increase in blood calcium level, they secrete calcitonin. So what does that tell you about the function of calcitonin? The original stimulus is going to be checked by the response. So if blood calcium increases, the function of calcitonin is to increase blood calcium more or to decrease blood calcium. What is it going to be doing to blood calcium? Increase, decrease, no change. You got it. Does that make sense to everybody? Its compensatory response is to decrease blood calcium. It's going to make you pee out more calcium. It's going to stop calcium reabsorption in the kidneys, and it's going to store calcium away in the bone. That's all AMP1 stuff. Opposite would hold true. Decrease blood calcium. Now we're looking at hypocalcemia. And now we have a different target cell. This is going to be the parathyroid gland. That's the target for that guy. So we'll be talking about that in just a second, but we'll cover it with this particular lecture point. And it secretes parathyroid hormone, PTH. And what do we know about PTH now? If the stimulus was hypocalcemia, what's PTH going to try to do to blood calcium levels? Increase, decrease, no change. I nod my head back and forth. I nod my head back and forth. Nod. You guys with me? Lovely, just nice simplified flow charts. Is this helping anybody retain this information a little bit better for everything? I mean, it's just so condensed. It's so powerful. Always think about the stimulus, therefore how the body is going to respond. If you can do that, physiology becomes easier. Not easy, but definitely easier. So here we are. Parafollicular cells secrete calcitonin. We know that the stimulus for calcitonin is an increase in blood calcium, and we know its response is to bring that blood calcium levels back down again. Parathyroid glands, boom, 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 boom. There's 4 little nodules on the back of the thyroid gland itself, on the posterior aspect. These secrete the hormone PTH. Within the parathyroid gland, we find the chief cells, and the chief cells are the ones that secrete that parathyroid hormone. We now just did a quick review. We know that the stimulus for the release of PTH is a drop in blood calcium levels. Therefore, we know PTH is all about increasing those blood calcium levels back up. It's going to cause bone to be broken down. It's going to stop the reabsorption of calcium in the kidneys, and it's going to cause the release of calcitriol, vitamin D3 from the kidneys, an active form of those, which does all of the above, plus it allows for calcium retention from your diet. So that's all AMP1 stuff. I will not test you on that part again, but you will potentially be tested just on this nice, short and sweet, make sure you understand calcitonin and PTH has to do with calcium regulation, okay? Is that clear? Is that fair enough? Beautimous. Those are simple reflex arcs for those that need to be able to answer homework questions. All right, pineal gland, this secretes melatonin. Now, melatonin is an interesting beast. There's a lot of misunderstanding about melatonin. What do most people already understand about melatonin? Well, they know that melatonin has to do with sleep. So how many people here have taken melatonin to help them sleep? This is a common one. Go ahead. If you have, if you want to, disclaim that. Yeah, me, yes, yes, yes. So it's not, this is, yeah. Weird fact, melatonin has nothing to do with sleep per se. It keeps getting used as a therapeutic modality for diseases that are associated with sleep, but it actually does not, it's not part of your sleep cycle per se. So what melatonin is, it is a hormone that tells your body that it's dark outside. And when it's dark outside, technically your body should be sleepy, but we're humans. So it don't work like that. Okay, so melatonin only tells your brain it's dark. It doesn't tell you to sleep. Does that make sense? So when you take melatonin, it might make you sleepy, just depends on the person you are, or it might not make you sleepy. Just because your brain knows it's dark doesn't make you go to sleep. You guys clear on that? Here's the deal with melatonin, though. The most important aspect of melatonin is that melatonin controls almost everything else in your body because melatonin helps regulate your circadian rhythm, and your circadian rhythm helps drive almost every other hormone in your body, which is going to be, circadian rhythm dependent. So that's kind of all I want you guys to know about melatonin is that it has a very powerful regulatory control on the rest of the body because it helps control how much hormone is being secreted at any given time. So even something like cortisol, which is your stress hormone, can be regulated by your circadian rhythm. All right, quick review. Adrenal gland histology. You got your capsule. Okay, that's this, when we zoom in to these striations, that's this dark blue layer, that's the adrenal capsule. And then what you remember about the abbreviations to learn. What's the most superficial layer of the adrenal cortex? Yeah, good. So what is the abbreviation, you guys, that I asked you guys to go ahead and try to learn? Yeah, let's just do the abbreviation. That was a good point by Andy to do that. So GFR, GFR, GFR. G, F, and R are the adrenal cortex. The capsule is not part of the uh, um, adrenal cortex. Sorry, I was reading Melissa's have to spell on exam. Not on the exam, but on the lab practical, yes, absolutely. Everything has to be spelled correctly, please. Especially with notes there in front of you. You know, you have the objectives list to be able to do your lab objectives with, to be able to do your lab practical with. So please, spelling is important. I can go into a whole spiel about how one misspelled word will kill your patient in the future, but you guys probably already know that. That's all it takes to misspell a drug or something, and then everything starts going awry. So this is zona glomerulosa, zona fasciculata, zona reticularis. That's the adrenal cortex. The rest of this in the middle is all the adrenal medulla. The medulla oblongata, no, just the medulla, the adrenal medulla. Each one of these areas secretes a different hormone. We discussed these last week. Everything secreted from the adrenal cortex is a steroid. Aldosterone, cortisone. Cortisol is the same thing as cortisone. Adrenal androgens. Androgens, that's testosterone. And then we can convert androgen, testosterone, into things like estrogen, progesterone, so forth, so on. Estradiol. Aldosterone is also known as a mineral corticoid because aldosterone's function, no, they are all androgens, Heather. It's just the primary androgen. Basically, sex hormones are considered androgens. Mineral corticoids, aldosterone. Remember last week I said you must know this, you must know this, you must know this about aldosterone. Can you guys tell me? I think I remember this. I could be wrong. And if I am, just smack me. I did go through aldosterone, right? If not, just tell me. Okay. And we'll remind ourselves on this because aldosterone's one we're not going to cover in heavy detail here in Unit 1, but it will be such an important part of your later, especially once we start getting into the renal system. But man, yeah, you guys are going to be tested heavily on aldosterone in the future. Heavily, like big 20, 30% of certain exams. So aldosterone is going to be an important one. So what aldosterone does, is it regulates minerals, right? That's why it's also known as a mineral corticoid, because the primary ions of your body are sodium and potassium. Make sense? These are minerals, these are ions, and they are pretty darn important to your body. And what hormone helps to regulate how much sodium and how much potassium are in your body? It's aldosterone. How does it do that? Aldosterone increases the amount of sodium that is retained in the kidneys. So the more aldosterone that is present, the more sodium retention we have. So what would happen to your blood sodium levels in the presence of aldosterone? Increase, decrease, no change. Hold on, work with me. Retention means to retain. So we're not peeing it out. That would be excretion. So what would happen to sodium retention? Well, aldosterone increases the amount of sodium we keep. Does that help? So that keeping goes back into the bloodstream. So in the presence of aldosterone, what happens to the amount of sodium in your blood? Increase, decrease, no change. Wonderful. Awesome. So how do we decrease the amount of sodium in our blood? We get rid of aldosterone. Does that make sense? And then there's also another hormone you guys are going to learn about that's secreted from the heart that causes sodium to be peed out. Now, potassium, we'll come back to this in pretty large detail, but in this particular case, aldosterone increases potassium secretion and excretion. So if we secrete it, then we excrete it, which means excretion, which means that it leaves in our urine. If we increase potassium secretion, excretion, the more aldosterone that's in the system, what's going to happen to your blood potassium levels? Increase, decrease, no change. Lovely. So aldosterone increases sodium in the blood and decreases potassium in the blood. It's pretty powerful, especially in a disease state. This alone can kill you pretty fast. Wouldn't the arrow go the other way? Help me understand. This is increased sodium retention, and this is increased potassium secretion. So these are two opposite things. If I would have said potassium retention, it would be an arrow down. You're not wrong. So it would decrease potassium retention, all while increases sodium retention. But the easier way, in my experience, the easier way to learn it is to increase sodium retention, all while increasing the amount of potassium that is excreted. Cool. All right. We are actually, you know what? We do need to review this, come to think of it, because it will be important for thyroid hormone. How would we be able to move sodium in one direction and potassium in another direction? What did you guys learn about in AMP1 that's moving so, oh my gosh. Oh my gosh. You got it. Lovely. You guys got this connection here, right? If I'm moving sodium one direction and potassium the other direction, well, that sure sounds like the sodium potassium ATP pump to me. Does that, you guys all on board with that? If we're moving stuff around, if we're retaining it, secreting it, no matter what, if we're moving ions around, we need a transporter for that function. What does that? Sodium, potassium. So really, when it comes down to it, aldosterone actually controls the amount of sodium and potassium pumps in your kidneys. That's how it do what it do. Loverly. Loverly. All right. Adrenal medulla. Again. Yeah, aldosterone with yeah. What would happen with that? It just depends on the patient, but often that's the whole idea of dialysis, right? Is to actually start getting aldosterone back to normal levels. Aldosterone is going to be freaking out pre-dialysis because all of those metabolites and wastes are building up in the bloodstream. That's the whole point of dialysis, to get that stuff out. Yes, no? So with aldosterone, unfortunately, you know, for the patient, there's definitely going to be a lot of fluctuation. It's a steroid hormone, so it sticks around in the bloodstream for a long time. Yeah, that stands to reason to me. Yeah, if they miss a dialysis, they were already hyperkalemic. Wait, actually, aldosterone levels should be elevated probably if they're hyperkalemic. Now, I'm just spitting this out. I don't actually know the answer to that because again, I've never done dialysis. Disclaimer on all that sort of stuff. I just use physiology to make a lot of assumptions. But one of the best response to hyperkalemia is to secrete more aldosterone because, check it out, right? What's the function of aldosterone? It helps us pee out more potassium. So if we have too much potassium in our blood, hyperkalemia, then we need more aldosterone to pee out more potassium. But that's just physiology. Now, you know, every single patient, especially if they're on the dialysis, it means they're not healthy. So, you know, if that's exactly how their body's going to respond or not, I don't know. It just depends on the patient. Does that make sense? But an assumption, the assumption is that they would be hyperaldosterone if they had hyperkalemia, especially chronic. All right, let's move on. Great question, Alyssa. Great connection. Let's move on to this adrenal medulla right here. The adrenal medulla is going to play a big role in stress-related function. What sort of stress? Axeman. Now, that being said, I have to make sure to reference that the adrenal medulla can function in normal everyday activity, but not very often. Okay, the adrenal medulla is triggered by extreme emergencies most of the time, and it's part of that sympathetic reflex. So this is the guy that secretes adrenaline. Adrenaline is a combination of epinephrine and norepinephrine. It's produced by those chromaffin cells that we talked about last week. And we just get this huge rush of adrenaline, epinephrine and norepinephrine combined. What do you guys think the function of epinephrine and norepinephrine are? Think about it in the terms of energy that we read over and over. Yeah, increase heart rate for sure. But I want you guys to be thinking about the energy connection. Just one more time. We're not done with epinephrine, norepinephrine. I'm going to teach you all the cellular products of what's going on with epinephrine and norepinephrine. You guys are going to be thinking about what it does to heart, what it does to blood pressure, what it does to urinary excretion. There's a lot to deal with. But in terms of energetics, do we want to increase the amount of energy available, or do we want to decrease the amount of energy available? You guys are fantastic. Keep going, keep going. Increase, increase. I need more. Lovely, lovely. So what do we know? We know lipolysis, we know gluconeogenesis, we know glycogenolysis, we know increased protein catabolism. We're going to be breaking things down to make more glucose available right now, because survival is the most important thing. See? Now, here's the problem with us as human beings. Does anybody ever feel like this? Yeah, I hope it's not constantly, but if it is, my apologies. College is tough, life is tough. Always try to find a release. Even when you're here, you know, in a summer course, try to find something fun that can get your mind off of school every once in a while. It's important not to focus on your worries, because when you focus on your worries, you feel like Mr. Axeman's chasing you. Could be your bills, it could be kids, it could be in-laws, it could be politics, it can be anything nowadays where you feel like Mr. Axeman's always chasing you. And when that happens, the sympathetic nervous system becomes hyperactive. And you're always going to have this low level of epinephrine and norepinephrine that is circulating through your body. So let me ask you, what's that going to be doing to glucose availability during that time? Increase, decrease, no change. You're stressed out. You're in this just mode of damn. Next is going to be another hormone we're going to talk about more, because we got more slides on it down here, but it's cortisol. Cortisol, most people know as the stress hormone. True story, if you've heard of cortisol before, you probably associate it with stress. Yes, no, maybe so? So cortisol has gotten a bad rap. Cortisol is essential for life, and it's the number one hormone in your body that helps you during stress. But what did I say earlier is the number one stressor on the human body? Not your bills, as crazy as that may sound, not your kids, as crazy as that may sound. The number one stressor on your body is hypoglycemia. Your body just has to fix this problem. But we can also tie hypoglycemia in with bills, right? Because if you're stressing about bills, your energy demands start to increase, and we start to use the glucose that's in our bloodstream. Therefore, we have a low level of hypoglycemia, and we need to bring it back up. So when you're stressed about whatever it is in life, and if you're chronically stressed, you're going to have epinephrine and norepinephrine being released, and you're going to be having cortisol being released. Function of cortisol is to increase glucose availability. Function of epinephrine and norepinephrine is to increase glucose availability. This is great. If we have a short-term running from Mr. Axeman response, we now have enough glucose to fire through our body and help make this ATP required to get away. Yes, no, maybe so? This is an alarm reaction. That first initial trigger of especially, you know, if it's like politically driven stress, you know, we all get that first initial trigger. Now, the body, and you know, it's kind of interesting to think through these, because we've all felt this. The body initially has tons of energy. But what's going to start to happen as that body is releasing glucose, and then it's like it's going to be starting to shout out, I have very little left. I'm running out of fumes. I'm running out of fumes. I'm running out of fumes. The stage of resistance is going to occur through your body. Exactly. This is that burnout feeling. And then finally, you get to this stage of exhaustion where you have nothing left. That even though you're stressed and your body's trying to elevate blood glucose, if you have nothing else, nothing left, you're fatigued, you're depressed. You just feel like you have to sleep all the time. You guys all know what I'm talking about, probably, because I think we've all been here before. It's a normal body reaction because we have killed it. Does everybody know what I'm talking about? I hope some of you maybe haven't, but unfortunately most people have. This has to do with hormones in the body, and there are ways that people can shut this stuff off, like exercise. The more stressed you are, the more exercise you can do, and that actually causes the body to release other hormones that will end up controlling epinephrine, norepinephrine, and cortisol. Exercise is a wonderful saver for a lot of different hormonal systems. Cortisol is all about stress, all about stress. So what do we know? Gluconeogenesis. Oh, great. Protein catabolism, protein degradation. Let's break down those amino acids so we can make them into glucose. By the way, what organ is the organ of choice for gluconeogenesis? Always the bridesmaid, never the bride. Yeah, keep it going, keep it going. What organ, what organ, what organ? Good old liver. Good old liver. Stimulates lipolysis. Now we're releasing fatty acids and glycerol. The glycerol goes to the liver for gluconeogenesis. So proteins going to the liver, amino acids going to the liver for gluconeogenesis. Inhibits glucose uptake into most of the body. Why? Let's get that glucose to the brain. Unfortunately, this is why we end up feeling fatigued and depressed when it's chronic. All right, pretty cool, huh? Do you guys see how learning those words last week has made this week a little bit easier, a little bit faster? As long as you understand gluconeogenesis and glyconeogenesis, or glycogenesis, so forth, so on, it will help you. So take your time to learn those words, and everything else will start coming together. Now, cortisol does have other effects. For instance, cortisol is permissive. It permits other hormones to do their jobs. Please write this down. I always test on the permissiveness of cortisol, always. Permissiveness, it permits, the root of the word is right there, it permits other hormones to do their job. That's why it's essential for life. Vasoconstriction is when blood vessel diameter gets smaller. We constrict a blood vessel. And if we're constricting a blood vessel, we have a blood vessel with a certain amount of blood going through it, and we constrict it. What's going to happen to the pressure being pushed against the walls of that blood vessel? Increase, decrease, no change. And if we increase blood pressure, what's going to end up happening is it's going to change the amount of blood that's flowing through that blood vessel. So without cortisol, we cannot have adequate vasoconstriction to certain areas of the body when we're running away from Mr. Axeman. That could be very, very bad, and it's going to actually cause people during a fight or flight response, if they don't properly vasoconstrict, what's going to end up happening is that blood's going to go areas, exactly. Blood's going to go to areas that you don't want it to go, and it's not enough blood's going to go to the brain, and you're going to pass out when you're running from Mr. Axeman. When you're running from the hippo or the lion, you're going to pass out. Sounds pretty evolutionarily disadvantageous, huh? Oh, there's a lion. Drop down to the ground. You just become food. So you're probably not going to last long out in the wild. Yes, no, maybe so? You don't need to understand it perfectly. I will not test you on that. I'm just making sure that you understand this whole permissiveness. There is another permissiveness of cortisol, and cortisol is required because it's permissive for glucagon to function. Glucagon is a hormone released from the pancreas. We will talk about it more tomorrow. We did talk about it last week when we said that pancreas has the islets of Langerhans with alpha cells and beta cells, and they secrete glucagon and insulin. We'll talk more about that again tomorrow. So don't stress if you don't remember that now. But without cortisol, glucagon can't do its primary job, and then you die. And then you die. Certain death. Anti-inflammatory. Sweet. It helps reduce inflammation. Wonderful. I love that. That sounds magical. In fact, cortisol has been used for over 100 years as a way to inject patients with, blown knees, back problems, elbow problems, joint pain. You inject those joints or muscles with cortisol, and it reduces inflammation in that area. Yeah, it's a miracle worker, kind of. Got to be careful. It is a steroid shot. Cortisone is a steroid. Anti-inflammatory response. It's great. It's wonderful until it starts being abused. And for the longest time, we didn't understand cortisol quite as well. Patients were getting cortisol injections way too often, and it was causing hypercortisolism. Hypercortisolism. What do we know cortisol even does? Stimulates gluconeogenesis, inhibits glucose uptake, stimulates protein degradation, stimulates lipolysis. So all of a sudden, these patients started having some pretty big issues with energy availability. Does that make sense? Because we are using it as an anti-inflammatory. By the way, the military still uses cortisol quite often. Athletes. So there's a lot of abuse of cortisol as an anti-inflammatory. Last but not least, immunosuppressive. It's an immune system suppressive. It suppresses your immune system. It stops your immune system from functioning properly, for Pete's sake. Let me ask you this. Anybody that has been in school at all, whether it was high school or college or whatever, during finals week, the stress is building up, you get done with all your exams, you're about ready to go party, and then all of a sudden you just feel like shit. You get sick. You're coughing, got a sore throat, strep throat, whatever the case may be. I don't know. It happened to me almost every single semester. Cortisol is an immunosuppressive. It breaks down your immune system. So if you're chronically stressed, are you more likely to get sick or less likely to get sick? There you go. Got to be careful with cortisol. People that are stressed out about things are definitely more likely to get sick. All right, let us take a break. We're going to be walking through the last of cortisol today as part of our lab. And then we're going to save insulin and glucagon for tomorrow. Then we're going to talk more about thyroid hormone synthesis tomorrow. Then we'll do some case studies tomorrow, and it'll be a lot of fun. And today for lab, let me just double check. We are going to be doing the safety, if I remember right. You guys should have already done the safety quiz, but I'll give you guys a few moments to do that. And then I'm going to actually be teaching you a little bit about heart anatomy today in lab as well. So, all right, let's take a decent break. Let's come back at, What time do we got? How much time? Sure. Let's come back at 50 after. Go get some coffee, bio, banana bread. Yeah. You want to say hi? No, too bad. Maybe it'll make you smile. Can you say hi? Don't want to, not right now. I think he's a gigantic present right now, wrapping himself up in present paper. That'll do it. You wipe your. Just don't tell him what you did. I don't think he wants your boogers on his back. Oh wait, maybe he does. Ew. Ew. So just a minute. Their mom is. I think she's done the laundry real quick. Anybody else at home with their kids? That is an amazing present wrapping right there. It's so good. Just my dog. My dog is somewhere, but I think he feels a storm coming in because he's starting to hide. And that's what he does when there might be even any chance of lightning. He feels it beforehand. Nice, Melissa. You want? Yeah. Hey, do you want to sing "Yum and Them"? Yeah, sing. Yum yum, num num and ho. Right, num num ho. Yeah. Oh, all the way to Arizona, holy. All right, Beckett. All right, just a couple more minutes, and in terms of just take a breath. I know I need one. Choo! Got you, right in up. Schnogger. You want down? Say down, please. Please, of course. There you go. Once they're out of my office, we can get rolling again. What are you doing? Everybody's throwing stuff at me. What's going on here? Am I a playground? We got Aniston, which is the little girl's name, and then Beckett, which is the be silly, be kind, be sleepy boy. Is it doing its thing? Why does it feel like it's raining on me? Why are you throwing at me now, Aniston? Are you copying your brother? Crazy kids. Sorry guys, just bear with me. Oh my goodness, just another second. Okay, you guys are. What? Hey, do you guys want to learn all about cortisol? I'll take that as a no. My son's actually pretty good with anatomy already, if you can imagine. When the quarantine first hit, I brought a lot of the models home with me, and he'd spent hours just looking at the models, and he started asking me, "What's this? What's that?" So he's pretty good with it all. All right, there we go. Say bye-bye. Bye-bye. Come say bye-bye. No, don't throw something at me. Say bye-bye. Bye-bye. Go to mama. All right, Beckett. There you go, kiddo. Here you go. Yeah, just a gigantic mess you got to clean up later. Can you close my door, please, Beckett? Sorry, 1 sec. I got you, bro. Here. Yeah, I understand. That's why I did that. All right, good. Last few things, then I got to get to work, guys. I want to get these guys going so they can be done. Nobody likes to sit and watch the rest of the world go by here. Let's go. Let's go. There you go. All right, have a good flight. Man, the peace and quiet of being home with your kids. Oh, wait, there is no peace and quiet, is there? All right, off my schnogger here. Okay. Nice. It's always good to have a kid that's just, they understand what, is going on in the real world. So spays and neuters and just real life happens. We should always be aware of it. Okay. Actually, I'm not going to do this. Thank you. Per se, I am going to come back to this, but we're going to write this down just like we always do. So I'm going to go back to this right here. We're going to go ahead and go to a new page because I don't have enough room here for it, but we're using the exact same flow chart to understand cortisol and cortisol release. So we just go ahead and write down our template. What is our template? Stimulus talks to the hypo. I'm going to start abbreviating it because I can. You guys can follow me, I'm sure. Hypothalamus to the AMP-PIT. AMP-PIT to a target organ, target cell. And that's going to end up with some sort of response, release of a hormone that can actually trigger a response. So we're going to go ahead and say response. And then what do we know? We know that the response is always going to be doing the opposite of the stimulus, and it does that by negative feedback loops. For cortisol, which is what we want the final response to be, the primary stimulus for cortisol release is stress. But again, what's the number one stress in the human body? Hypoglycemia. Body doesn't like it. Hypoglycemia reduces the amount of energy availability, and the whole body runs on energy. So this is a pretty big deal. This is a pretty big deal. Now, that being said, what we're really dealing with is stress. But stress is a little bit subjective. Try to get you guys all online, linking it all together with hypoglycemia. Now, this stress is going to trigger the hypothalamus to now release one of these RHs. But in this particular case, which type of RH are we dealing with? Just give me the abbreviation and then we'll work out the whole word. Good, Ashley, right off the bat. That was nice and fast. Final story is cortisol. So this is a corticotropin releasing hormone. Just like TRH was thyrotropin releasing a hormone, now we're dealing with cortisol, so it's corticotropin. And this travels the couple of centimeters down to the anterior pituitary. It has receptors for CRH and will secrete what in response? ACTH. Adreno, as in going down to the adrenal gland, right? Going to the adrenal cortex. Adrenal corticotropin hormone. So it's going to be going down to the adrenal glands and causing the release of cortisol. Let's get it down to those adrenal glands. It gets into the bloodstream. It goes everywhere, but where do we find receptors? in the adrenal cortex. But remember, the response is cortisol. And what part of the adrenal cortex did we learn secretes cortisol, cortisone? Yeah, so there was a specific zone that did that secretion. The G, the zona glomerulosa, that was aldosterone. GFR, the R, those are the sex hormones, the gonadotropins. And then the F in the middle, that's cord. So this is going to be, the target cells are actually zona fasciculata of adrenal cortex. Now, if you remember that or not, doesn't matter. It's written down now. Does that make sense based on everything else that we've learned? Just a quick Y would be helpful, or N would be helpful, because then I can go back. Awesome. Zona fasciculata, once stimulated, secretes cortisol. And of course, cortisol has a negative feedback loop. It's going to try to inhibit the release of CRH if there's too much cortisol. But what if there's not enough cortisol? What's that going to do to CRH? Increase, decrease, no change. If cord levels are low, and we want everything just right. Talks back to ACTH. These are complex reflex, multiple integration centers. Good stuff, good stuff. Now, cortisol, if the number one stimulus is hypoglycemia, what's the function of cortisol? Just use your logical skills to work yourself through that. Based off of what we based off of what we learned about growth hormone and thyroid hormone and everything. You got it. So if the stimulus is hypoglycemia, then the response is to bring that blood glucose level back up. Does that make sense, everybody? So what we're going to do, kind of ran out of room down here, so I'm going to just draw an arrow this way rather than down, because you won't be able to see as well down there. And I'm going to say increase BGL. B stands for blood, G stands for glucose, L stands for levels. This is how it's written out in medical practice. BGL, blood glucose levels. Either have high blood glucose levels, low glucose levels, or normal glucose levels. And we're always relating it to the blood because the blood's the transporter of glucose. So the function of cortisol is to increase blood glucose levels. Okay? How do you guys feel about that flow chart? Probably much more simple than it would have been if we would have started last week on it. I hope it makes sense. I hope it kind of stands to reason. Does cortisol do more? Of course it does. But does everything that it do relate back to glucose? Yes. Except for the anti-inflammatory and immunosuppressant properties of it, of course. So let's go back here real quick, and you'll see that that's what we're dealing with. Stress is also controlled by a diurnal rhythm. So that's circadian rhythm. So what hormone helps control your circadian rhythm, everybody? Yeah, melatonin. So melatonin's going to play a factor in cord release. It's pretty cool. Its function is to increase glucose availability. So check it out. Increase blood glucose, gluconeogenesis, glycogenolysis, you name it. Increase blood amino acids, which means protein catabolism, protein breakdown. Why are we breaking down proteins? What are we going to do with these amino acids? Just party with them? What do we do with the amino acids? Good, Andy. That's exactly the word I was looking for. Does it make sense, everybody? If we break down proteins, we get amino acids, they go to the liver and they make new glucose. via gluconeogenesis? Yes, no, maybe so? What about lipolysis? Where are these fatty acids going? Remember the fatty acids in the glycerol. Glycerol is going for gluconeogenesis, but where are the fatty acids going? This was the PTSD event last week. They are going into the blood, you're not wrong. But what's the function of all those free fatty acids at this point? What are they used for? Hooah! What are they good for? Let me just throw down a quick reminder of a slide that we did last week. Where do those fatty acids go? The glycerol goes to the liver for gluconeogenesis. Where are those fatty acids going into? Was this a helpful little poke, a little prod, a little reminder? Those fatty acids are going into the Krebs cycle to help with the production of ATP. Okie dokie? Okay. Not something I necessarily like have a test question on, but it is an important aspect of biology. We, along with all the other cells of the universe around us that we know of, require ATP. And a very large majority of cells utilize the Krebs cycle for that function. And therefore, a lot of cells require fatty acids to help run that Krebs cycle and glucose. Okay, back to it. I hope this all makes sense. We summarized this big block as, goodness gracious, increase BGL, but on a test, please don't just write BGL. This is just a summary, right? You need to be able to tell me how that blood glucose level is increased. Fair enough? We're just summarizing. We're trying to put as little down as possible so our brains can retain the information as best as possible. The more you write, the harder your brain has to work to retain that information. We all feel that? Anybody ever been in those, you're sitting down, you study, you write down a paragraph, and then you leave for 60 seconds and you come back and you don't remember anything you wrote down because you wrote down a huge paragraph of crap. Am I the only one? I think that's most students. We write too much when we're studying, and that is a big problem. A big part of my teaching style is try to help you guys find new study techniques that work. For the large majority, I promise you, the less you write, the more you learn. And that's what flowcharts are good for. The less you write, the more you learn. You link it all together. Again, cord is diurnal. So what you'll notice about that, di means two times a day. What you'll notice is that it peaks in very specific times of the day. It's basically every single 12 hours, but it's just to help regulate those blood glucose levels at all times. Here is another one of these wonderful flow charts that has diagrammatic and understanding of how everything works too. Stimulus, time of day, and stress. Those are the primary stimuli, just diurnal, where you can't really do anything about cord levels. During certain times of the day, you're going to feel a little bit more anxious. You're going to feel a little bit more on edge. And that has to do with those cord levels, or partially has to do with those cord levels. Stress, though, will be the number one stimulus that I test you on. Causing a release of CRH from the hypothalamus, causes the release of ACTH from the anterior pituitary, which now goes down to the adrenal glands, but more specifically into the adrenal cortex and the zona fasciculata. Now, one more thing to think about is cortisol. What classification of hormone is cortisol? What classification? Steroid, biogenic amine, or protein, peptide? Good. So as it's produced, how is it going to get out of those zona fasciculata cells? Is it going to be actively transported, or is it going to be passive? It's passive. So let's just think about it. No, you're all right, Alyssa. That's why I asked these questions. Can a steroid just pass through the cell membrane without help? As long as it can pass through a cell membrane without help, that's called passive. It doesn't need help. It doesn't need a transporter, so it's not an active process. Does that make sense? So as cortisol is being made, it's just going to go from an area of high to low. So it's going to go from the zona fasciculata. It's not stored here or anything, and it's going to just directly secrete out into the bloodstream. Cool. Well, what do we know about it? Yeah, but we'll get back to thyroid hormone. We haven't covered everything about thyroid hormone yet, Heather, but absolutely it needs a carrier protein. In fact, a lot of people are hypothyroid, not because they lack making thyroid, but because they lack the carrier protein for moving thyroid around. But anyways, yeah, we'll do the same thing here. Is if this is nonpolar, if this right here is a steroid, it's nonpolar, can cortisol dissolve in your bloodstream? Yes or no? The answer is no. So what does it need in order to be transported through the bloodstream? It's right here in front of you. Yeah, exactly. A carrier, a carrier, a carrier. Wonderful. And in order for that to happen, that carrier protein is called cortisol binding globulin, CBG, cortisol binding globulin, or otherwise known as corticosteroid binding globulin, otherwise known as cortin, all sorts of different words for it. And guess what? What organ produces over 90% of all of your blood proteins? So again, if somebody has a liver disease, what might be a result to their CBG levels? Increase, decrease, no change? The answer would be decrease. So the liver disease means that the liver's not functioning as well. Does that make sense? So the amount of CBG would drop if the liver can't produce it, can't synthesize it. The amount of CBG available would be dropped. And what would start to happen downstream? Are we going to have something that looks like hypercortisolism, or are we going to have something that looks like hypocortisolism? Is it going to look like there's too much cort, or is it going to look like there's too little cort? Hyper or hypo? Remember, the carrier protein is required to move that hormone to where it needs to go. So if I don't have the carrier protein, what happens to the amount of cortisol being transported? Increase, decrease, no change. Hold on, stop. No, don't re-listen. Just listen again. Sorry if I'm not making myself clear. CBG is required to transport cortisol downstream. If I have somebody with liver problems, potentially their CBG levels are dropped. There's less carrier transporters. There's less Uber, right? And if I don't have as much Uber, what's going to happen to the amount of cortisol that can be transported? Increase, decrease, no change. Alyssa, great question. Pretty easy to answer. Why does it need CBG if it's passive transport and nonpolar? Passive transport and nonpolar just means that it can leave the cells of the adrenal cortex and it gets into the bloodstream right here next to the adrenal cortex. But if it does not have CBG, it can't get to the brain. It can't get to your fingertip. It can't get to your heart. It can't get to your small intestines. It would be stuck here. It can't be transported once it gets into the bloodstream because it's non-polar. Oil and water don't mix. Does that make sense? It's going to be scared. It has a hydrophobia, right? It's scared of your bloodstream. So it's going to run back into the tissues around the adrenal cortex. It literally is going to leave the bloodstream again if it doesn't have an Uber that picks it up. It's going to be so scared, it's going to run back out from high to low. You're welcome. But did that make sense to everybody? If I have less CBG, it doesn't mean I have less cortisol, but I'm going to have less available cortisol at the target organs, and it's going to look or appear like hypocortisolism. Yes, no, maybe so? These are the lessons I want you guys to learn about physiology. It's not necessarily, I'm not teaching you pathophysiology, I'm teaching you what normal function is, and therefore what you can predict to happen in patients in the future. It just makes medicine a little bit easier. What's going on downstream? Man, CORT has all sorts of effects. It's going to end up causing the release of glucose from the liver, making new gluconeogenesis, causing more fatty acids and glycerol to be released from adipose tissue. Let's break down proteins to release amino acids because those amino acids are needed for gluconeogenesis and other things as well. And if CORT levels get too high, that's where we get the anti-inflammatory effect and the immunosuppressant effect. That was not good at all. And worse, the last worst part of this is that you got to be careful with CORT injections, because just because you have an anti-inflammatory that reduces the pain, it doesn't make it heal faster. Kind of a bizarre combination, right? You're making it so you're less painful, but it doesn't mean that you're going to be fixing the problem there. Increase retention of sodium. This is going to come back to aldosterone, but we'll deal with stress and stress effects on aldosterone in a later chapter. All right, that's where we're going to stop with the endocrine system today, and we're going to move on to lab. Are there any questions about what we covered today at all? Whether you started the homework or started working through some practice review stuff, you know, I'm available for questions on all of those. Good, awesome. Don't forget, you guys already have a test coming up this weekend, so be on the ball, be on the ball. Homework, when is homework due? No, not today, Alexis, thank goodness. No homework due today. Birthday exam, sorry, Heather. Yeah, so your Unit 1 homework will be due Saturday at 6 p.m. Does everybody remember that from last week? It will be consistent through pretty much the entire semester. We'll always try to have the exams due Saturdays, except for Unit 4, because that kind of falls within the finals week. So we can't do that for Unit 4. The rest of the units will all be the same. Homework will be due Saturday at 6. And when is your lecture and lab practical going to be due? When is your lecture exam and your lab practical? Sunday at midnight. So please follow this. This is an important deadline. Know it. If you're even remotely concerned about these due dates, text me. I don't want anybody missing deadlines. They're you know, a big part of your grade. The homework's worth 50 or I guess 40 points. 40 points. So if you miss it, that's 4% of your grade gone. 4% just disappears. That's a big chunk. Easily make the difference between an A and a B or a B and a C. So please make sure you get it turned in. You could turn it in today, for Pete's sake. So there's no reason why you have to wait until Saturday at 6 p.m. to turn it in. Is that clear? Okay. And one last thing, I had a lot of questions over the weekend about due dates, which is great. If you have a question, text me. But don't forget that I gave you this schedule right here. And I'm going to be as responsible as possible that if you want like follow this schedule, it will always tell you when things are due. So for instance, what's due today, June 7th, that's the endocrine anatomy group study guide. One per group and it's due today at midnight. Does that make sense based on everything that you've been taught to this point? What's going to be due on Wednesday? The cardio anatomy that we discussed today, it's going to be due at midnight. June 10th, blood anatomy study guide and a hematology lab report. So we'll always have things kind of following these deadlines here. Study guide, yeah, study guide is extra credit. So they're going to be all of the anatomy. Study guides are always anatomy. That's right, because you weren't here for the first day. So that's why you're probably, yep. So anytime it says anatomy group study guide, those are extra credit. That's your way to get extra credit through the semester. I will always give extra credit for you guys just doing what you're supposed to do anyways. You should be making study guides of all the lab objectives. So you'll see from today what we do, and your lab group will get you further up to date on this as well. Let's see here. There was one mistake on this printout. What was it? It was down here. Remember, I know somebody put this out in the group me as well, but I had two people that saw these due date discrepancies. Thank you for seeing them. I thought this was the first semester in a long time that I hadn't made a mistake, but turns out I did. Unit 3 homework due on July 10th. Well, the exam's always the day after, so Unit 3 exam and lab practical will be July 11th, not 18th, okay? Please make sure you make note of that. Unit 3 lecture exam and lab practical are July 11th, not the 18th. Is that clear? Awesome. Okay, let's see here. We will be using this some more. Get my coffee over here. What we have to go through now is heart anatomy. Let me show you guys where to go to get all this in case you've forgotten. So just go to your Blackboard tab. You go to the lab material tab right here. You go to the labs for Unit 2. For Unit 1, I don't know why I clicked on Unit 2 because I'm crazy. And you'll see right here is heart anatomy. I've put quite a few links on here. One of them is to YouTube that I put together. So what I've done is all I've started to put together are some YouTube. These YouTube videos are for the entire semester. I have a whole bunch of different YouTube videos. You can go to my, I mean, I don't have very many subscribers because I don't ask you guys to subscribe and I don't promote it. I don't care. You guys can use it to learn or you don't have to use it to learn. There are other ways to learn as well. But if you go here, you'll see there's some of me right here going through nervous system and muscles and respiratory system, respiratory anatomy. So I have quite a few of these YouTube videos for you guys to watch, especially as the semester goes on and the system gets even crazier. But cardio stuff is all here. So I have everything for all of your cardio physiology, which is Unit 2. I have everything. I got blood flow, cardiac output, conduction pathways, autorhythmic action potentials, like every single bit of detail that you guys want to know for your exams is right here in these YouTube videos. You can see it's the exact same format as your class. It's all chalk talk. So it's very helpful to have that connection, I think. One second, Heather. We'll get there. I promise. So if you guys want my YouTube channel, here it is. By no means, there's no pressure. I'm not out YouTube for money. I just want you guys to learn. You guys know that, right? I hope. I only did this because I was forced to with the pandemic. I don't even know how to use this most of the time. So this is going to be what we are covering today. This is the exact same spiel that I'm going to be giving today is blood flow through the heart. We want to look at the heart. And as we look at blood flowing through the heart, guess what you're going to be learning? You'll be learning the anatomy. Does that make sense? Because blood's going to have to flow through very specific chambers to get to where it needs to go. And there you go. There's your cellular respiration. Why do we care about that? That sort of stuff. So this video is going to have all of that information of blood flowing through, out to the lungs, back in. And this is what we will be covering today. So I hope you guys can use it to understand the anatomy of the heart as well as the physiology of the heart. It should be useful. Now, there are also some links to something that you have available because you are an ACC student. One second. I'm just showing you everything that's available for today. If you, yeah, pretty please. You guys all have so many good questions, but I promise I'll get to them. Well, I think I promise I'll get to them. I hope I promise I'll get to them. What's going on here? Due to security reasons? I didn't know that it decided it was going to do that. Huh. Gale 3D. So maybe I'll just do this. Gale Interactive Library, Anatomy. Here it is. I don't know why I did that. So in case you guys didn't know about this, which you probably didn't know about because most students don't know everything that ACC has available to them, there's something called a Gale Interactive 3D model. So with this 3D model, you can go in, you click on Systems, and then with this, you can actually click just on what you want. Nervous, skeletal, systems going on here, show more result. I don't know why it doesn't always filter very well, but there we go. Blood supply, circle of Willis, all these sorts of things you're going to learn more about later. So it's pretty cool in terms of that, that you can actually click on this stuff. Now, where sometimes you have to do like some serious searching. Yeah, all of this you could have used for A&P1. Obviously, there's cranial nerves and all that crazy stuff, joints. Here, heart anatomy. So heart anatomy, you just kind of, I'm sure there's other ways to search for it, probably just up here in search. But this will be helpful for today's lab because you don't have a model in front of you. I mean, you can't, you're sitting at home. So using this particular extension, you can actually dissect the heart and you can look at all of the different parts of it. You can right click and you can move the heart in a three-dimensional way. So you can actually see the back of the heart and the front of the heart. And then you can dissect pieces away from it as you go. So it's pretty cool that way, right? Over on the right, you're going to have some text that'll tell you a little bit more. Same material that we cover in class. It's all here. All of it will be here. You can go through structural terms and it starts to label them for you. So it's a pretty powerful tool. And again, it's free to ACC students. So I highly recommend that you utilize it. All of these, tricuspids, papillaries, right ventricle, left ventricle, interventricular septum, these are all going to be terms that you guys learn today. Anybody like going, how did I not know this existed before? I don't know. I always find that students don't know this exists, and it's it's free to use. You don't have to buy a $50 app for your phone to do your anatomy. Okay, let me see if this link will get you to where you need to go. There we go. Here's just to find the heart, since my link through Blackboard is not available. Okay, back. Okay, next. You're welcome. Next, cardio lab models unlabeled. So you click on this, that'll pull up all of these pictures of models that are not labeled. And this is what you guys can use to do your extra credit study guides. Just pick a picture, any picture. You don't have to label every single one, but find your lab objectives on these and try to label them in as many different views as you possibly can. You have a lot of pictures available to you, somewhat overwhelming. I know, I understand, but that's the responsibility of a student, sorry to say, is to go through and weed out what you want to use and what you don't want to use. So the last little bit of information before I go into the hematology quiz thing is that, so Melissa was like, what should we use in order to study today's lab? Well, boom, that's right here, right? That's just the heart anatomy. Where the heck am I? Heart anatomy. So although spring 2017, the objectives haven't changed since then. So just open up the PDF or the doc. You can print off that Word doc or just look at it as PDF. These are the objectives that you guys need to know. Identify these structures right here. Everything else is from sheep heart dissections. And then you'll have to identify the same structures that you found above. You just have to find them on a sheep heart. It won't be that hard. Not that long of a list of objectives, right? Relative to some of the other ones, like nervous system from A&P1, super long list, right? Looking at a skeletal system, muscles, all that sort of stuff. So you guys all know what you need to be able to label, what your objectives are, yes, no? So now you go into these pictures of the models and you find your objectives. And you label it. Like, find a picture that shows an epicardium, find a picture that shows a myocardium and an endocardium. We'll cover these terms, don't worry. Exactly like the endocrine study guide. I'm not changing it at all through the entire semester. So every single anatomy lab that you guys have, I give you a list of, or a PowerPoint full of pictures. You guys will label them for a study guide. I don't make you search the internet for model pictures. I do it all for you. Fair enough? You guys just have to label because it's part of the learning process. See? Oh, no. You guys rock. Thank you. You guys rock. Oops. I want to click on this. I want to click on this. Give me 12 more seconds. Maybe 15. Stop counting. I'll get there. Let me click on this. Trying to see if I have a file that I want to share with you guys. If I do, then great. If not, then I'll be sad. Looks like I do. Lovely. All right. So I will also upload this. This is by a professor, Joan Hauser. She does a number of these. I've worked with her on these as well. They are great. So I'll also make this available to you guys. This is a drag and drop. So the drag and drop activity, you have a bunch of arrows pointed at different parts of the model. And you just take and say, where is the aorta, where is the apex, so forth, so on. So these are your lab objectives down here, and you just have to find them on these different models. And you just switch your position of view, you find them on that model. You switch your position of view, you find them on that model. Very helpful, and we will actually cover a bunch of this together today as a group. These are not turned in as part of your extra credit study guide unless you want them to be a part of it. You know what I mean? But these by themselves won't cut it. It does not correct you, Melissa. No, this is literally just a PowerPoint. You just drag and drop, literally make it go where it needs to go. So if you have any doubt about where something is, just holla. Just give me a holler and I'll make sure that you know what you're supposed to know. Okay, fair enough. We'll be doing a little bit of this today. I'll go ahead and drop that in. In right now as we speak. I will add it so that I don't forget it later, which is something I would do. Definitely known to do that. Let's see, heart, drag and drop, submit. So that'll be available for you guys to use ASAP, right? It's right there now. There's the D&D heart on diaphragm D&D activity. Sound good? You guys all feel like you have everything you need to study this material? And now we're going to go through a spiel. So I'm not leaving you guys to fend for yourself. We're going to go through a spiel now. Everybody feel like you have what you need? Yes, no? Give me a C, give me a no. If not, just let me know and I'll try to figure out how to help. But a few of you did a really good job over the weekend. You're like, is this what I'm looking for? Or here's a model, Dr. H. I don't know what the hell I'm looking for on this model. And I just took a picture, edited that picture, drew a circle around what they were looking for. I labeled it for them and I sent it right back. So I'm here to help. Don't think that you have to do this on your own. Okay? Physiology's hard. So hematology lab safety quiz. The due date is tonight at midnight. That can be found in the exams and practicals, Heather. So because it is like a quiz, and it's right here at the top, hematology blood lab safety quiz. You guys need to have this done by tonight, pretty please. You just hit begin. It's very easy, right? Minimum penalty for failure to follow hematology safety procedures is that, blah, blah, blah. If you have this condition, you should not participate in the hematology lab exercise, blah, blah, blah. And what you'll notice, again, I'm not trying to trick you guys, not on this, is that if you go to the lab material for Unit 1 and you open up the PowerPoint that I gave you, the hematology lab report PowerPoint right here, slide 1 answers pretty much question 1. Slide 2 pretty much answers question 2. You just go through each one of the slides and there's a question on each one of those slides. Plus or minus, there's a little bit of discrepancy, but for the most part it works like that. Does everybody know that they have a hematology lab report quiz due today? And where it's found is in exams and practicals. It's on your schedule as being due today at midnight. So just follow that schedule and it'll always lead you right. Okay, we're finally ready to go. We're finally ready to go. And what we're going to do is we're going to get started by thinking about the heart and thinking about its function. So, and we discussed this last week briefly, but if I had to describe the function of the heart in one word, what word could we use? What is the function of the heart? Yeah, it's a pump. And what's it a pump for? It's a pump for blood. Good. So we're going to go ahead and say heart. The heart is a pump. It's not just any old pump. We'll get back to that. But the heart is a pump and it's a pump for blood. It pumps blood. Perfect. The heart is actually a dual pump. It's not just a single pump, it's called a dual pump. Dual. Which one is the fight and which one means two? D-U-E-L, is that fighting, or D-U-A-L? I never remember. Dual. Which one's two? Dual. Thank you. Thanks, guys. So there we got ourselves a dual. Yeah, that makes sense now that you wrote it out, Alyssa. Perfect. Dual pump. It's a double pump. How does this work? There's a right side of the heart and there's a left side of the heart. Each one of those is a pump. The right side is its own pump and the left side is its own pump. The right side pumps blood to the lungs. Blood out in the lungs is called pulmonary circuit. We'll come back to that. The left side of the heart pumps everywhere else. It pumps all of this blood out to the rest of the body. So the heart definitely is a pump, and it pumps blood, but it doesn't just pump blood every time the heart beats. It doesn't just pump all the blood to the same spot. It's a dual pump, meaning that as it pumps, the right side of the heart pumps blood out into the lungs, and the left side of the heart pumps blood out to the rest of the body. Is everybody with me on that? I actually didn't know that when I got into physiology for the first time. Some of you probably already knew that, but I always want to make sure to reiterate this because I didn't know it. I'm not ashamed to say it. When I was 19 years old, I didn't know this. I just thought I'd pump blood, and I even have a heart condition, and I didn't know that. So there you go. The next thing to understand is related to cellular respiration. What did we learn about cellular respiration? We learned the formula. And we said in cellular respiration, cells of the body need oxygen and they need glucose in order to make ATP. But waste product is CO2 and water. True story? And then last week, we also made sure to mention the fact that oxygen is this beautiful non-polar molecule. There's an oxygen double bound to another oxygen. This thing is very, very non-polar and tiny. What does that tell us about its solubility? Is it water soluble or lipid soluble? Yeah, this thing's actually lipid soluble. What? Wait a minute. Okay, well, if it's nonpolar, can it dissolve in the blood? Yes or no? Nonpolar. This is a fat in essence. It's oxygen. It acts just like a fat. So what does it need in order to be transported through the blood? It needs a Uber. Yeah, it needs an Uber. Perfect. It needs a carrier protein. Not a vesicle. Vesicles wouldn't be able to hold on to nonpolar things, Tariq, because vesicles are made phospholipid bilayers. Therefore, non-polar molecules would just whoop right through that cell membrane. So it needs a carrier. And what is the carrier for oxygen? Sabrina and Brianna have already answered that. So what is the carrier protein for oxygen? Hemoglobin. So oxygen is transported because it binds to a carrier protein called hemoglobin. Binds to hemoglobin. Everybody with me so far? Because it's nonpolar, it needs a carrier protein. What is that carrier protein? It's hemoglobin. Now, hemoglobin is actually made up of 4 smaller protein units. Each one is a particular type of globin. There's alphas and betas. And in the center of each one of those is a heme group. And the oxygen actually binds to this heme. Heme. What heme is indicating in this particular case is this tiny little molecule the body makes. I'll teach you more about it in a later lecture about blood, but this heme group at the very center is iron. Fe3+. We got an iron chilling in the middle of this thing. Some of you may have known this, but many of you did not probably know this, is that iron and oxygen are sweethearts. They are like just fastidious lovers. They they have to be together. Oxygen will do anything it can to find iron, and it's going to bind onto it as tight as it can. It doesn't always win. Okay, oxygen. If you went to a park that oxygen and iron had both been to, you'd find this written in the tree, right? Etched, emblazoned in the side of that tree. Oxygen loves iron. They heart each other. Oxygen, heart, iron. They're cute together. As long as they're together, they make this beautiful red color. Okay, when oxygen binds to iron, to your eye, the way light reflects off of it, it reflects back as this red color. Romeo and Juliet. Yeah, that's right. Okay, we got it? Oxygen loves iron. It wants to bind to iron, and when it does, we get this very beautiful red color. Think of, a rusting car on the side of the road. Car, metal, made out of steel. Steel has iron in it. So as steel rusts, it turns like an orange-red color. True story? Basically, Hook'em, Texas, UT colors. This weird burnt orange color. Well, without the presence of the other minerals, if it's just iron, it's red, scarlet red. Let's put it this way for physiology's sake. If oxygen is bound to hemoglobin, what color will that blood be? Red. Now, as the hemoglobin starts to drop the oxygen off at the tissues, it's got to say its farewell. It's a horrible, horrible heartache for that iron. Well, now that iron has another job. Really, it's not the iron at this point. It's the hemoglobin itself. It's this globin group itself. It starts to pick up CO2. And CO2 loves the globin. In fact, CO2, it's really interesting, and I'm not going to deal with it too much, but if I got a big globin group, just imagine the circle's the globin group, and the iron's right here in the middle. Well, unfortunately, CO2 is bigger than O2, and so CO2 will come in here, and it binds right over the top of the iron group. So can oxygen bind to the heme as long as CO2 is present? Oh, that's sneaky CO2. That's dad right there saying, no, you can't date my daughter. That's what's going on right there. So CO2 binds to that globin group. Now, CO2, when it binds to this globin group, it causes it to turn a purple blue. CO2 bound to hemoglobin is a purplish color, a blue color. Now, most of the time, there's still always oxygen bound to your hemoglobin. No matter what, there's, typically still 70, 80% that's bound to hemoglobin. So your blood is always a little bit red. But what do we know about venous blood? If you are, Caucasian skin like me, when we look at the blood return to the heart, what color are these blood vessels in my arm? They're a bluish purple color, right? And that's because it's not deoxygenated. I'm going to teach you guys never to use that word again. I don't like that word, and there's a reason why. But there is poorly oxygenated blood returning to my heart, and therefore that blood is a purple-blue color. Does that make sense? Real simple idea. I just trying to make a game out of it. I want you guys to understand it, that as long as oxygen's bound to your blood, bright red, and as long as CO2 is bound to your blood, it's blue. Make sense? Thumb up, thumb down? Why is this so important, Dr. H? Why are you doing this to us? Let's go back here. If I'm pumping blood out to my lungs, well, what's the function of the lungs? Anybody want to try to answer that? What's the function of the lungs? Oxygenates O2 in, CO2 out. A little bit more clear than just oxygenates the blood. It's actually, the answer is called gas exchange, which means O2 and CO2. Just a quick side note, your body actually cares more about CO2 than oxygen. So it's something we've been mistaught our whole life until now. But if the function of the lungs is gas exchange, the function of the lungs is to get rid of CO2 and to get O2 in. What's the blood going out to the lungs? Is it going to be poorly oxygenated, or will it be heavily oxygenated as it's moving towards the lungs? Yeah, so what color will the blood be as it's moving out towards the lungs from the right side of the heart? Blue. Purplish, you got it. And what color will the blood that's pumping out to the body be? Because from the right side and from the lungs, what this blood actually does is it oxygenates, it goes back to the left side of the heart, and then it goes out to the rest of the body. Awesome. Now we have ourselves red blood, and all the models you look at from here on out, maybe this will help you. I got blue blood vessels and I got red blood vessels. What do we know about blue? Don't say vein, don't say anything like that. Just tell me about its oxygenated state. If it's blue, is it poorly oxygenated or oxygenated? Don't say CO2 either. Let's go poorly oxygenated or oxygenated. Beautiful. And if it's red, poorly oxygenated or oxygenated? Perfect. Was that helpful just for an initial glance at this model? When you look at things like the right side of the heart, they kind of color it a bluish color to help you. You look at the back of the heart, you got this purple blue, poorly oxygenated. We know that this is going to have something to do with poorly oxygenated blood, most likely going out to the lungs-ish, but this is coming back from the rest of the body, superior vena cava, but going into the right side of the heart. And what we know about the right side of the heart is it's associated with the lungs. So blue, blue, and more blue. Cool. I hope that helps you guys tie some things in together as we go through these models. Loverly. There was another question up here that I skipped over. Oh, it had to do with poorly oxygenated versus deoxygenated. That's what it was. So why do I not like the word deoxygenated? If your blood was deoxygenated, you would be dead. And in a lot of textbooks, they use this term deoxygenated. And the fact of the matter is kind of what I was mentioning earlier, is that your blood tends not to drop below 80% saturated. So almost all the time, your blood has like 80% of the hemoglobin retains oxygen. Even when it's returning back to the right side of the heart and out to your lungs, it's still mostly oxygenated. Does that make sense? I can't remember who wrote deoxygenated, but does that make sense to everybody? It's never deoxygenated. And it's been a term that gets thrown around in textbooks and stuff, and just for me, it bugs me. So try to avoid using it, because I think it makes it so you literally are learning the wrong thing. You're thinking that there's no oxygen in the blood going back to the lungs, and that ain't true. Anybody ever been hooked up to a pulse oximeter or had your kid hooked up to a pulse oximeter, right? When you're looking at these pulse oxes, they basically just clip it on your finger and using these values here, they're able to determine the partial pressure of oxygen in your bloodstream. If it's even if it's below like 95%, they start to freak out. If it's not 98%, that's not a good thing. So as you start to get partial pressure of oxygens down here at 93, they think you're pretty darn sick. And what that means is that only 93% of your blood has oxygen bound to it, and yet they're freaking out. So it's mostly oxygenated. It's 93% oxygenated, but they're still worried about you. Does that make sense? So never deoxygenated, always pretty heavily oxygenated, in fact. And if it drops below around 85% oxygenated, this is called hypoxia, hypoxia. So no bueno. All right, cool. We got that done. I could tell you right now, it's pretty fast and easy. The pulse oximeter has a specialized laser that shoots through it, and the laser is it's pretty darn easy, yet it's a little complex. Oxygen refracts light differently than CO2. So it has to do with the measurement of the refraction of light, laser light. So as the laser is refracting, depending on how much oxygen is present, you'll get a specific amount of refraction. It's really a simple little lesson in physics right there. And on the opposite side is a laser detector, and you just line it up. The laser goes through your bloodstream and refracts light in a very specific pattern depending on how much oxygen is present. Pretty cool, huh? You're welcome. Okay. Here's what we're going to do. We're going to learn the heart, but we're going to learn the anatomy of the heart using a flow chart. We're not even going to look at a damn heart to begin with. Why, Dr. H? Why? Another question for you guys about study habits and stuff. Who here, when they have a model in front of them, spends all this time looking at the model, trying to find a particular lab objective, and they search all these internet images to try to find where to do that lab objective, and then they take and they go, okay, here on this model is this lab objective. They'll label it with a piece of tape or whatever, or here online, you know, with an arrow. And then you just move on to the next lab objective. And by the time you get 2 or 3 lab objectives away, you've already forgotten what that first lab objective was. Anybody ever done that? That was me in a nutshell right there. Is I was so focused on finding the word on the model that I never learned the word. Does that make sense? Who here has English either as a second language or has learned another language? Maybe they're bilingual, maybe not even fluent, but just any languages whatsoever. And when you're learning a new language, you don't have every single word sitting in front of you in the physical form. You know, it's not like you have an airport sitting in front of you when you're learning the word airport. All you do is learn the word. And you learn these big lists of words, and then you have to associate them by using them. You can't learn a language unless you use the language. True story? Yet why do students think that in anatomy, all they have to do is have a heart, have a list of words, and then it should magically all come together? It's not going to unless you learn the list of words and then use those words in context. That's when you learn. It's just like any other language. Stop treating it like a lesson in school and start treating it like a language, and it'll change your guys' life and how fast you learn this. So objectives. Oops, oops, oops. Here's your list of objectives. You guys need to learn base, apex, the different layers of the heart, epi, myo, endo, right atrium, right auricle, right ventricle, left atrium. And if you're just looking through this list, nice. Nice, great, Tariq, I love that. Perfect. And then you'll have that study guide for when you do the lab practical, especially since it's open note, right? Now you got it, it's right there. Should make the lab practical that much easier as long as you're learning as you go. Anyways, just like last time, do you guys remember? I mean, as I'm saying these words, auricle, ovalis, fossa ovalis, interventricular septum, you know, sometimes it's in one ear, out the other ear, where it's like just a bunch of gibberish, just like histology for all the endocrine system, just a bunch of gibberish. But the more we saw pictures on them and the more we used the words, you guys were able to start to follow along. So let's do that again, but with the heart. And the best way to do this with the heart, it's a flow chart. Dr. H and his flow charts. Dr. H and his flow charts. So what we're going to be talking about is blood flow. Just like in my YouTube video, it's all about blood flow through the heart, because blood's got to flow through the heart. So blood flow through heart. Again, the heart is a dual pump. The right side of the heart pumps where, everybody? Where does the right side of the heart pump to? Yeah, to the lung. So is the right side of the heart going to be poorly oxygenated or richly oxygenated? So what color will it be? The right side of the heart. It's pumping blood out to the lungs. It's going to be, so I got a couple people that just were a little bit mixed up, and that's probably because words are not always easy to follow. Tariq, you already corrected yourself, and Sabrina, you know, I'm not calling anybody out. I just want, if just a couple of people get it wrong, it means more people are confused by it. So just a reminder, the right side of the heart is a pump. So look at my hands. It's got fluid inside of it. It's got blood. So think of a water balloon. As this water balloon contracts, what's going to be happening to the pressure inside of this chamber? Increase, decrease, no change. And if there's a fluid in there, it's going to help that fluid move to the next chamber. Does that make sense? So the right side of the heart is pumping to the lungs, not backwards, always to the lungs. There's going to be some doors in the way you guys are going to learn about that prevent it from backflow. But as it contracts, it's sending blood to the lungs. Well, if the function of the lungs is gas exchange, then if it's before the lungs, it hasn't gone through gas exchange yet. So it still needs to get to the lungs to drop off CO2 and to add oxygen. And if it still needs to drop CO2 off, then it's still blue. Does that make sense to anybody that might have been confused? The right side of the heart will be poorly oxygenated, bluish blood. True story? And did that make sense to anybody that might have been confused? The right side of the heart is poorly oxygenated, bluish. And so when I draw my flow chart out, guess what color I'm going to be using to describe the anatomy of the right side of the heart? I'll be using blue. It just helps me understand my flow chart that much better. All blood enters the heart through two huge veins. Ooh, we haven't done one more thing. So, lo siento, let's just do this quick definition because students get this wrong all the time. Let's define artery and let's define vein. So what's the definition of an artery? Anybody want to give it a try? Just a guess. Oh, dang you for being so right off the bat. Dang it. Yeah, good job.
Why am I saying dang it? Because how many of you guys out there thought artery means oxygenated blood? It's okay if you did, please. I'd like to at least get this nipped in the butt at least a little bit. If nobody thought that, spectacular. But a lot of students get it wrong because they think artery means oxygenated blood. It means red. Just because you look at a model, thank you, Heather, just because you look at a model and you see a blood vessel is red, it doesn't mean it's an artery. Because the definition of an artery is any blood vessel that's moving away from the heart. That is the definition of an artery versus a vein. It's always towards the heart. You'll see now when we start going through this blood flow that some arteries will be blue and some veins will be red because they're going out to the lungs and back to the heart from the lungs. So, a little bit bizarre. Anyways, all blood enters the heart through these big, huge, cavernous veins called vena cava or vena cava. Okay, and there is both a vena cava on the top, and in Latin and medicine, above is superior. So I have a superior vena cava, and I have a vena cava down below, which be an inferior vena cava. All right, so I have a superior vena cava and I have an inferior vena cava. You could draw it out any way you want. You can change the text so that it works for you. For me, since I know this so well already, superior vena cava, I understand to write these words together in my head. Inferior vena cava. All blood enters the heart through either a superior vena cava or an inferior vena cava. And as it enters the heart, the first chamber it's going to enter is the entryway. In an entryway to like a mansion, I'm not sure any of us have a mansion. If you do, congratulations. But those big, huge, entryways is called an atrium. Like as you enter their atrium, bizarre word, but that's exactly what it is in the heart as well. The entryway to the heart is called an atrium. So the first chamber it enters is an atrium. But this atrium is on a very specific side of the heart. It's on the right side of the heart. Why? Blue, right? It hasn't made it out to the lungs yet. So we're returning poorly oxygenated blood to the right side of the heart. And we have to make note of that. This is the right atrium. Make sense so far, everybody? Whether you know what the picture looks like, whether you know what the model looks like, it doesn't matter. Learn the words. The right atrium is a muscular chamber of the heart. The right atrium is a muscular chamber of the heart. And it's going to take, and as it contracts, and because it's gravity fed as well, blood is going to move from the right atrium towards the next chamber, the larger chamber. The larger chambers of the heart are called ventricles. But again, we're still on the right side of the heart. These are larger chambers because there's more muscle. They're going to be able to provide more pressure gradients, more muscle, the more strength of contraction we can have. But it's still on the right side of the heart. These are the right ventricles. Well, right ventricle, because there's only one of them. These larger muscular chambers are larger because they are going to be pushing the blood out into the body. So from the right ventricle, what did I teach you? Where is this blood going to get pushed out from the right side of the heart? Where is it going to? Is it going to the lungs or is it going everywhere else? Perfect. So this is going to end up in the lungs. But in order to take a relatively large volume of blood and to push it out towards the lungs, it's going to have to go into a pretty large blood vessel. Now again, this is still blue. But this large blood vessel, is it going to be an artery or a vein? As the blood vessel is moving away from the heart towards the lungs, is this an artery or a vein? It's not a vein. That's the trick, right? Away from the heart is an artery. It's all right, Andy. I'm glad you're doing it now, right? This is how we learn. We learn from mistakes. The right ventricle is going to be pushing blood out into a very large artery called the pulmonary trunk. And all trunks, gosh, I wrote trunk, pulmonary trunk. All trunks in the human body, there's a lot of trunks, brachiocephalic trunk, cephalic trunk, celiac trunk, so forth, so on. All trunks in the human body branch. That's what the terminology for a blood vessel that is a trunk is about, is it's going to branch, it's going to diverge, it's going to split. So the pulmonary trunk is a very large artery that carries poorly oxygenated blood to the lungs, but in order to do that, it's going to have to branch out to the right lung, and it's going to branch to the left lung. Does that make sense? It's going to go through a right pulmonary artery and a left pulmonary, I ran out of room, artery. We either have a right pulmonary artery or a left pulmonary artery, and all this blood is moving from a bigger artery branching into smaller arteries that go to each respective lung. And in those lungs, okay, in those lungs, it's going to feed into these huge pulmonary capillary beds. Okay, so both of these are going to be feeding into pulmonary capillary beds. And whether you know this or not, capillary beds, very small blood vessels that allow for gas exchange and nutrient exchange. So these pulmonary capillary beds is where gas exchange is going to be occurring. So as the blood passes through them, it drops the CO2 off and picks up oxygen. So what color am I going to be changing my flow chart to now as I move away from the capillary beds? Perfect. So these pulmonary capillary beds are going to start to now return oxygenated blood back to the heart. Now, are these going to be veins or are these going to be arteries? If I'm returning back towards the heart, yeah. So even though that's red, because it's oxygenated, it's still a vein. How bizarre is that? I know, it really confused me when I was first learning all this, but it wasn't until I just stuck to the definition. Boom. These are pulmonary veins. There's a lot of there are actually 4 major pulmonary veins, but you guys can just clump them all together into pulmonary veins. There's a cluster of 2 pulmonary veins coming from the right side, and there's a cluster of 2 pulmonary veins coming from the left side, so there'd be 4 total pulmonary veins. But the pulmonary veins return to the heart. So they're going to go back to an entryway of the heart. So those pulmonary veins are going back to an entryway. And in Latin, what word are we going to use as an entryway? Welcome to my house. It's going to be an atrium. Perfect. So this is an atrium, but now we are on what side of the heart because our blood is red? So this is a left atrium. Wonderful. Now the left atrium is also a small muscular chamber, not as big as a ventricle, but it's still a small muscular chamber. And as it contracts and is gravity fed, blood is going to feed into the larger chamber. So what's the larger chamber of the heart called? It's a ventricle. It's a ventricle, but oxygenated blood. So what side of the heart are we on? This is the left ventricle. It is a left ventricle. And the left ventricle is even bigger muscle than the right ventricle. We'll come back to why that is. It's a very thick, big muscular chamber. Wow, man, it's going to produce so much force, and it's going to squeeze around all that blood, and it's going to be able to produce a ton of pressure. And as it does, blood, as that heart contracts, blood is going to just eject. It's going to erupt like a volcano out of the heart. Just right on out of there. And it's going to enter a huge artery that exits that heart called the aorta. You will learn about the different parts of the aorta, the ascending aorta, the aortic arch, the descending aorta, thoracic aorta, abdominal aorta. It's freaking huge. But the important thing about the aorta is we got this big, huge, elastic blood vessel that a lot of blood's going out into, and now the aorta gets to help with the initial transport of the blood to the rest of the tissues of the body. The rest of these tissues of the body are called the systemic circuit. This is the brain and all the rest of the organs of the body. Anything that's not the lungs is systemic. Over here would be called the pulmonary circuit. This is the systemic circuit. But in the systemic circuit, it's going to make it out to systemic capillary beds. And then from the systemic capillary beds, what do we know about capillary beds? What's happening out in those capillary beds? What's happening out in those capillary beds? Wonderful. Good job, Alyssa. Does everybody remember that? That's where gas exchange occurs. So what's happening to the oxygen? It's going to get dropped off. And the cell is going to use the oxygen to make ATP. And what are those hemoglobin groups now going to be picking up? It's dropping oxygen off and picking up CO2. So now our blood's going to turn blue, and it's going to end up returning all of this blood, depending on where it's coming from, back into the superior and inferior vena cava. Back into the right atrium, right ventricle, pumped out the pulmonary trunk, out into the pulmonary arteries, out to the lungs where gas exchange occurs. We pick up oxygen, return it to the left atrium via the pulmonary veins. It's going to move from the left atrium to this big, huge muscular chamber called the left ventricle. Blood gets ejected out into the aorta to your whole body. Gas exchange occurs. Circulation, y'all. So let's just answer some quick questions. Does everybody have this written down? Just trying to get you guys to quickly use words. Okay, just use this flowchart to answer the following questions. Blood enters the left atrium via what blood vessels? Perfect. Blood enters the right ventricle from what chamber? Wonderful. Good. Good. I know not everybody's the fastest typer. I hope if you're not typing, you're still kind of saying it out loud or thinking it out loud. I'm okay with that. Everybody's not the same boat, and that's okay. Blood enters the aorta from what chamber? Just fantastic. So let's do something. Let's go to some model pictures. Just trying to find some real good ones. So what if I told you that this big open chamber right here was the right atrium? Look at your flow chart. If I have a right atrium right here, where do we know all blood comes from in order to enter the right atrium? Go ahead and write it down in chat. If I have a right atrium, where's blood coming from? And what color should that blood vessel be? If it's the vena cava, what color should it be? And I could either have a big blue blood vessel that's above it. What would that be? Superior or inferior? And so here I am. I have a right atrium with this big blue blood vessel right above it. So what do I know this big blue blood vessel on this model must be? How do you guys feel about learning the word first and then applying it to the model? Was that helpful? It's a really weird concept that, you know, I know it helped me as a student. It might not help all of you, but it certainly helps me, and I try to relay that on to you guys. So now if I have a right atrium and I have a chamber down below it right here, the model's been cut away, this chamber down below it, what chamber do I know blood is moving from the right atrium into the? Got it. What if I told you, so this is a posterior view of the heart, a little bit more complicated to see right now, I know, but what if I told you that this, where my cursor's at right now, if I were to cut that open, that's where the right atrium is found. So a big blue blood vessel that is below, because this is a posterior inferior view of the heart, so this would be below the right atrium. What blood vessel would this one be right here? Good. Does that make sense to everybody that that would be the inferior vena cava? Perfect. If not, let me know, let me know, let me know. There's another term that you guys haven't learned yet. It is in your lab objectives, and I want to point it out. Do you guys see how there's something that looks like big dog ears or elephant ears? Boom, 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 boom. Right here and right here, kind of funky looking ears. Yes, no? So in Latin, the word for ear, you might have learned it in A&P1, for your actual ear that's sitting outside your body, it's called an auricle, an A-U-R-I-C-L-E, auricle. Do you guys remember that term? If not, no worries. Maybe you learned it as pinna, I don't know. But this is an auricle. The auricle are the ears. They're the physical external structures. There's a right auricle and a left auricle. Now, if I cut this auricle open, there would be a chamber inside of that auricle. If I were to cut that auricle open, there's a chamber inside of that auricle. So the right auricle has what chamber inside of it? Ah, the right atrium. Does that make sense? So one of the things students get wrong all the time is I'll put a pin in the auricle, say for instance this auricle, and they write as their answer left atrium. That's not accurate. The auricle is the physical ear structure. The chamber on the inside is the atrium. Are you guys clear so you don't make that mistake? The physical structure on the outside is an auricle, and the chamber on the inside is an atrium. Please make sure you get that distinction taken care of. All right, cool. So now if I have a model like this, I have two large muscular chambers down below. Remember, everything's always in anatomical position. So the larger muscular chambers, what do we call these things combined? What are the large muscular chambers of the heart? ventricles. You can't see the atrium because they're inside of the auricles on these models. Now, anatomical position. Which chamber would this side be? If it's coming from the right, there's the superior vena cava. Inside of here would be the right atrium. That's going to be the right ventricle. And then what did I say about the left ventricle? Does it have more muscle or less muscle? So that's a helpful little guide. So when you're looking at models and you see a chamber that's just massively muscular, it's got to be the left. Why? Why is the left side of the heart more muscular than the right side of the heart? Anybody want to guess? Yeah, if it has to pump out to the entire body, does it need more pressure to do that job or less pressure to do that job? Is everybody on board with this idea? The more muscle there is, the more pressure can be created? Yes, no? Lovely. Good. Okay, awesome. Let's go back to our flowchart. And this is what I'm trying to teach you guys, is don't just use models. Don't just use flowcharts. Use the combination of the lab objectives, the models, and the flowcharts, and you'll find yourself learning. Like magically learning, not memorizing, but learning. And hopefully your college career will just kind of make a little bit more sense. All right, it's always the hope. I don't know, doesn't always work, but that's kind of my goal. Now, we do not want blood going backwards, ever. These arrows are very, very important. The arrow points to the right. The arrow doesn't point to the left, the arrow points to the right. And in order for blood to always flow to the right through this heart, we need some doors that will stop the backflow of blood. We need doors that help blood move only in one direction. In order to understand this, yeah, we'll come back to the valves, but in order to understand this, I just want you guys to think about your own doors that are on a hinge. If I have a door that's on a hinge, and that hinge only allows that door to open this direction. If I were to push back this way, look at the door to the room that you're in right now. I don't know whether it opens into your room or out of your room, but mine opens into my room. So if I were to push on my door, I wouldn't be able to open it, right? Does that make sense? I'd have to pull on my door to open it. Well, this all has to do with pressure gradients. And then normally in a classroom, I'd show you guys using an actual door. It makes it pretty easy. But imagine a door that's opening up in a direction. And on one side of the door, you have an elephant. And the other side of the door, you have Dr. H. And that elephant is pushing back in this direction. Okay, that elephant is just pushing back this way, but the door only opens this way. Am I going to be able to put enough force onto this door to open it if there's an elephant pushing against me? Yeah, there's not a chance in a lifetime. There's no way I'm going to open this door, because why? If the pressure here is greater than the pressure over here, then that door is going to stay closed. Does that make sense? But what if I recruited a tractor? What if I made it so that the pressure here was greater than the pressure over on this side? What's going to happen to this door? Open or close? If I were to able to put so much pressure against this that I beat the elephant, then this door would open. Does that make sense? And that's the exact same way that your heart works. Is that your heart it has doors, and those doors are called valves. And there's valves in 4 places of your heart. There's a valve between the right atrium and the right ventricle. There's a valve between the right ventricle and the pulmonary trunk. Let's get to the left side of the heart. Well, if there was a valve between the right atrium and the right ventricle, I hope it stands to reason that there's going to be a valve between the left atrium and the left ventricle. And last but not least, there was a valve from the right ventricle to allow blood to leave the heart. So I hope it stands to reason that there's a valve from the left ventricle that's going to allow blood to leave the heart. So there are 4 valves of the heart. 1, 2 separate the atrium from the ventricle. 1, 2 separate the ventricle from the circuits. If it goes from the atrium to the ventricle, these are often known as AV valves, atrioventricular valves. So there's a right AV valve and there's a left AV valve. Okay, well, there's another way to learn them. It has to do with the number of cusps that these valves have. Best way to look at that is via this, right? So you can actually see that these valves have these little flaps on them. They kind of, it's still not perfect in these models, but each one of these has what are called cusps. They could either have 2 cusps or they can have 3 cusps. So these are called bicuspid and tricuspid valves. Bicuspid and tricuspid. So how do I remember where they are? So there's either going to be a tricuspid or a bicuspid between the right atrium or a right ventricle. So the way I remember this is an old school adage. We don't do this as much anymore because Amazon, but anybody ever been to the mall and you were taught like by somebody that you always try before you buy? Always try it before you buy it, right? I don't know if you've learned that or not. It's a little bit less as a representation of our kids nowadays, but you always try before you buy. And that's how you remember the valves. Always try before you buy. So the valve between the right atrium and the right ventricle would be the tricuspid. And the valve between the left atrium and the left ventricle would be the bicuspid. The bicuspid valve is also known as the mitral valve. It's often referred to the mitral valve in medicine. Doctors typically call it mitral valve. Normally, if you're having valve replacement, that sort of stuff, it's the mitral valve that they're talking about. Mitral regurgitation is blood that goes back from the left ventricle through the mitral valve back into the left atrium. So there's a lot of diseases of the mitral valve. The valve that separates the ventricle from the blood vessel leaving the heart, they're both called semilunar valves. Semilunar valve. Semilunar valves look like little half moons. So that's why they call them semilunar. Lunar has to do with the moon structure. So there's a semilunar valve on the right side of the heart, and there's a semilunar valve on the left side of the heart. So how do we distinguish between these? We just say what they're going to. So because the right ventricle sends blood out to the pulmonary circuit from the pulmonary trunk into the pulmonary trunk, we call it a pulmonary semilunar valve. Why did I write valve twice? I don't know. Why did I do that? I don't know. Lo siento. So this is the pulmonary semilunar valve. It's a semilunar valve sending blood out to the lungs. What about over here? I have a semilunar valve, but it's sending blood out into the aorta. So what am I going to call this? aortic semilunar valve. Does that make sense? Good. We'll come back and actually let's do it now. So if I go back to my models here, we'll go ahead and we'll look at this one. So here we are. I have a right atrium, we've already established that, and a right ventricle, we've already established that. And right here in the middle, this white stuff right here is a valve. These white cords are something different. I'll teach them to you in just a minute, but the solid white material is a valve. What valve, look at your flow chart, what valve separates the right atrium from the right ventricle? So if you can find your landmarks, you know where you're at. It makes it much easier. Let's go over to this model right here. So this model right here, now we have the left side of the heart. This is the left ventricle. This is the left atrium. So what valve separates the left atrium from the left ventricle? Perfect. Perfect job. Perfect, perfect, perfect. Now let's start looking at some front views of the same thing here. This is the left ventricle. This is the right ventricle. If I look inside the left ventricle, there's a valve that has these cords attached to them. This valve would be coming from this chamber above it. So what valve, again, am I going to be finding on the superior border of the left ventricle? Good, that's the mitral valve, wonderful. But what I want you guys to notice is that there's another valve right here. And this valve has these little semi-moon-like structures. 1, 2, 3, 1, 2, 3. Everybody see those? So which valve would be from the ventricle, the left ventricle, and leaving the left ventricle? Good, Beck. Good. So do you guys see that that would be the aortic semilunar valve? If you were to follow it, it would run deep and it would come right out here. This is the aorta. This is the ascending aorta, the aortic arch, portions of the descending aorta, and then these are 3 big arteries that branch off of it. So that is pretty cool. Now on this picture as well, on this side or on this side, now we can see the right ventricle. Now the right ventricle has, again, these cords are attached to the valve that's coming from the atrium. So if it's coming from the right atrium into the right ventricle, what valve is this? Tricuspid valve, always try before you buy, wonderful. And then what about this? This is from the right ventricle. It's going out into a big blue artery right here. So what valve is that going to be right there or right there? Good, that would be the pulmonary semilunar valve. By the way, what blood vessel would this be then? This big blue artery that's leading away from this pulmonary semilunar valve, right ventricle, boom, right there. What's that? You guys, good, good. Awesome. What else can we do with this? Quite a bit, actually, quite a bit. The right atrium and the right ventricle, the left atrium and the left ventricle, okay, they're kind of lined up. The atria, the ventricles, and the ventricles. And separating those two is a big wall, okay? So from the right atrium, left atrium, there's actually a wall that separates the atria from the ventricles. They lie in between the ventricles. This big, huge wall of muscle that you see right here lies in between the left ventricle and the right ventricle. So it's in between or inter. So this wall in between, and I'm going to go ahead and just draw it in as a line because I like lines in my flow charts, not other sorts of structures, but you can do it any way you want. If there's a big wall, goodness gracious, I'll just use the highlighter on the other side. If there's a big wall that separates these things, that wall is going to be called the interventricular septum. Septum means wall. You have a septum in your nose, right? Septum means a wall that divides a left and a right side. So septum, wall, interventricular, in between the ventricles, there's a wall. You guys feel okay with that terminology? All sorts of views of it, but it's this big, big, big chunk of wall in between those ventricles. Okay, awesome. Let's get back to the valve. Let's look at this picture to see these chords. These are your heartstrings. Anybody ever heard playing your heartstrings? These are your heartstrings. They're otherwise known as chordae tendineae. Yes, they're on your objectives list. See, chordae tendineae. They're tendinous cords that attach one side to the valve. The cords themselves, if I were to put an arrow in the cords, do not tell me that that's the valve. It's not the valve. Those are chordae tendineae. Is that clear? These guys here are chordae tendineae, not valve. Oops, I made the arrow go the wrong way. My bad. These are chordae tendineae. And do you guys notice that they're attached to this funny little shape underneath? You see that that is a distinctive shape of muscle. It's different than the walls. If I were to change the angle ever so slightly in a different view, I might be able to see it here. Yeah, there's a good view. There's something that looks right there. There's something that's pretty distinct, and the cords are going down and attaching to that. You guys with me that there's something a little bit different about the shape of that muscle there? On this model, the chordae tendineae now attached to that structure right there. Why am I using red on a, I'm so weird. Here, let me see if I can get that changed for you. Red on a red, that's helpful. Orange? Nope, nope, we'll go with the green. We'll actually just make it a bigger. That'll work. Okay, so that right there would be kind of part of it as well, although I'm not a big fan of the representation of this thing on that model. Shows it right here pretty darn well. Right there and right there. Both of these long projections. Do you guys see how they kind of look like a finger? They got this little nub, like short finger on this side, left ventricle is a little bit longer. All right, let's get to the gist of this thing, is that these are papilla. And in Latin, papilla means nipple. So you have papilla all over your body. You have papilla on your tongues. I think you only have one tongue. How many tongues do y'all have? You have papilla on your tongue. You have papilla on your skin. You guys learned about dermal papilla and hair papilla. You learned about those in the integumentary system back in AMP1. Papilla on your tongue, you learned about that in the sensory portion of AMP1. Here in AMP2, you're going to see the word papilla a lot as well, and it's these nipple-like projections, these small finger-like projections that protrude out into. Now in the heart, these are made out of muscle, so they're called papillary muscles. So anytime I'm pointing an arrow at these finger-like, nipple-like projections, I'm asking in the heart, I'm asking you about papillary muscles. And you can almost always tell the papillary muscles because, let's go down here again. Right there is a great indication of it. But do you guys see what's attached to it? What's attached to it right there? What am I pointing at? What's at the tip of the arrow? CT, good. You guys with me? What's attached to that? Chordae tendineae, good. And then on the far side of that, what do I have? I have the valve. Valve, chordae tendineae, papillary muscles. The purpose of the papillary muscles is these long columnar muscles that will contract and relax, and they help control the opening and closing of your valves. Because they're attached to the chordae tendineae, your heartstrings, they can help regulate when these valves are open or closed. Primarily, it'll be pressure gradients, but then the papillary muscles are going to help with that function. Does that make sense? All right. A few more words to learn. The very tip of the heart out here. The very tip of the heart out here. The very tip of the heart out here. You can kind of see that it looks a little bit like a whale snout, right? It kind of looks like a whale head and a whale snout out here. This tip of the heart is called the apex. This is the apex of the heart. The opposite side of the heart is where we find all these blood vessels here. Let me get this. Sorry. All right. Do you guys see how I kind of have a dotted circle over here, and it just shows a general area where all of these big superior vena cava, the aorta, the pulmonary trunk, right over here. This is called the base of the heart, which is bizarre. Students get it wrong all the time because they think base should be bottom, but that's not what it means. Base has to do with the anatomical representation of where everything is coming out of it. So the base of the heart is this side, and the apex of the heart is this side. Okay? Everybody clear on those two terms? Next, what do we got next? I'm just trying to do this all via memory. Oh, okay. Layers of the heart. Layers of the heart aren't too bad, honestly, as long as you remember a couple of terms from A&P1, and I will teach them to you in case you were never taught them. So, some terms from A&P1 that you may or may not remember. You guys remember like pericardium, endoneurium, yeah? Epimesium. So we had epi, we have peri, we have endo, all these crazy words. Now, if you could learn these words and actually learn what they mean, they're very helpful. Then you had mesium, so you had a root of MYS. You had neurium, so you had a root of neuro. You had chondrium, so you had a root of chondrio, or chondro. You had osteo, or ostium, right? Periosteum, osteo. And all of those were tying together, where you had an epimesium, or a perineurium, or an endochondrium. Yes, no, maybe so? Do you guys kind of remember this? I don't know if you were taught like this, but does that make sense in terms of words that you had in A&P1, whether you remember them or not? Hopefully there's a small memory of that. The last part of it is EUM. So that's the suffix of every single one of those words. Epimesium, perineurium, endochondrium. Eum tells you it's connective tissue. It's a layer. Eum tells us it's a layer. So perimesium, for instance, was a layer of your muscle. Epineurium tells you it's a layer of part of your nervous system. Chondro tells us cartilage. Osteo tells us bone. Yes, no, maybe so? So what about cardio? What does that tell us? So what if I had had epicardium? What does that tell us? Heart layer, but specifically, would it be inside, outside? What's going on? Yeah, it would be surrounding that heart. Does that make sense? What about endocardium? Inside. You guys are going to be learning other words this semester, like metria. Metria tells you uterus. So endometrium would be the inner layer of the uterus. Does that make sense? If you can learn how these words work together, you don't have to second guess yourself anymore going through the anatomy of it. You guys with me? Another word you'll see thrown into this mix quite a bit is myo. Myo, these both tell us muscle. Myo actually means mouse, little mouse, which is a little funny, but whatever. We end up using it as a muscle term. So myocardium, myometrium, what does that tell you about that layer? Is it's full of muscle. Yes, no, maybe so? All right, so let's go back to our model. Well, we'll actually just go back to our objectives real quick to show you the layers of the heart wall that you guys need to learn are epicardium, myocardium, and endocardium. IUM, EUM, same thing. It's EUM. It has a mouse. It's a little mouse. It had to do with how muscle moved underneath the skin. They thought it was funny that it looked like a little mouse rippling. It was weird. I don't know. Anatomists hundreds of years ago, they had to have their chuckle probably because they were high on mercury or something. So now you got the roots of these words. Epicardium. Where are you going to find this layer of the heart wall? On the outside, on the inside, in the middle? Where are we finding it? Endocardium. Where are we finding this layer? Outside, inside, middle? Let's go back to our model. What if I were to point an arrow right here? What layer of the heart would that be, man? It's pointed right in all this muscle, right in the middle. What layer of the heart would that be? Right in the muscle, right in the thick of the muscle, that's myocardium. Great job. What if I put an arrow pointed to the outside of the heart right here? What would that be? The surface of the heart is a very thin layer of connective tissue called epicardium. What if I were to put it in this tan stuff? I don't know, tan, what color is this? I don't know, I'm bad with colors. Peachy? Different colored stuff. Now, what do you have to do on the lab practical is you gotta read my questions, because my question will say, what layer of the heart is this? Don't answer left ventricle. Left ventricle is not a layer. Does that make sense? So read the question. The question will tell you what I'm looking for. And if you're in any doubt whatsoever, do you remember what I taught you last week? You can never be too specific for me. So you could answer endocardium of the left ventricle, and you will get full credit if you have any doubt. Is that clear? What structure is this? Papillary muscle of the left ventricle, and you will get it correct. I don't want you guys to get something wrong because you don't quite understand where the tip of the arrow is. Do your best, and if in any doubt on my question, just answer with very specific answer. And what I mean by specific is not just papillary muscle. This is, Remember, it's papillary muscle, not a papilla. You have mini papilla. This is a type of papilla called papillary muscle. Make sense? Sorry if I confused you earlier. Cool. Did I cover? I think I covered everything except for one thing. One thing. I think there's only one thing left to go through. And that is this interesting idea about fetal development. Now let's go back here. Everybody just stop. Don't look at anything. I should just stop sharing my screen here. Just stop, look away, think. Does a developing fetus breathe air? Are their lungs functional? They are in mom's uterus. Are they breathing yet? They're inside of a sac of fluid, right? So are they breathing, yes or no? The answer is no. I hope that makes sense. A fetus is not breathing. So where is it getting its oxygen from? It still needs oxygen. It still has cellular growth, division, coming from the blood, and that blood's coming from mom. Does that make sense? Is that a baby, a fetus, is getting all of its oxygen from mama bear. Mama bear gets a lot put on her. She has a lot of responsibility whether she knows it or not. All that baby's nutrients, all that baby's oxygen, all of that baby's waste, peeing and pooping right into mom. Well, not really pooping, but you get the point. Metabolic waste is going to be excreted by mama bear's blood. So mom's got a lot of work to do. So that being said, look at the heart. Do we need the blood of this fetus to go out into the pulmonary circuit in order for gas exchange to occur in a developing fetus? Yes or no? The answer is no. The answer is no, because baby's getting its blood from mama bear. Does that make sense? Will there still be blood that goes through the lungs? Of course, because the lungs are still developing. But is it going to be a massive amount of blood? Is it going to be all the blood for gas exchange? No. And the reason why is because in a developing fetus, or the reason how, I guess I should say, is because in a developing fetus, they have something called the fossa ovalis. And the fossa ovalis is a flap. It's a depression, right? You literally read the definition here. I'd like to see a picture of the fossa ovalis, or some of, that's a really cool picture right here. So in the, here we go. This kind of shows it, a patent foramen ovale. So in a fetus, there's something called the foramen ovale, because foramen just means whole. And blood, poorly oxygenated blood, well, you're getting oxygenated blood straight from mama bear. So there's really not a poorly oxygenated system going on here. Blood is going to enter the right atrium and immediately move to the left atrium to go to the left ventricle and get pumped out to the rest of that fetus's body. So there is a hole in between the right atrium and the left atrium. Crazy, huh? Just allows for blood to flow from one side to the other. You guys with me? So at childbirth, a membrane has been forming this whole time. And at childbirth, when that baby is born, due to the change in pressure gradients that occur, this fossa ovalis, which is this membranous material, is going to close over, and pre-fetal development, or sorry, pre-birth, it's going to start to form as well. And this is going to form over the hole. This is called the fossa ovalis. Now, if that fossa ovalis doesn't close completely, there's still going to be some holes that are present. If the fossa ovalis does not close completely, then there's still going to be some foramen that are present. Does that make sense? So post-childbirth, somebody that doesn't have the fossa ovalis form completely, they might still have blood going from the right atrium to the left atrium. Poorly oxygenated blood going from here directly over to the left atrium. How does that sound? This blood here will never get oxygenated because it can never go out to the lungs. It doesn't normally result in a lung disorder, although it can if it's long-term chronic. Yep, exactly. So that's all going to be sonography stuff that they are going to be able to determine this. Hopefully not surgery, post-mortem kind of thing. But here's one of the worst things that can happen is that if the hole is big enough, now all of a sudden blood clots that form later in life, those blood clots can move from one side of the atria to the other side of the atrium, straight into the left ventricle, and get pumped out to the rest of the body. So it's one of the not leading causes of strokes, but it definitely is a major cause of strokes because this improperly formed fossa ovalis is the #1 cardiac birth defect in the world. #1 is improper of cardio function. #1 is improper closing of the fossa ovalis. Pretty crazy, huh? And when you're looking at your model, what you might start to notice is that there's a big circle as you're looking into, it's kind of here on this model. It's not the best model for it. It's right here on this model, but that's not a great model for it either. One second. Let me see. I promise I'll show you guys how I typically test on this. Aha. Now, what students will often get into a bad habit with, it can be seen here too. So this model shows it really well. This is the exact same model you just saw online. You guys see that big circle right there? That would be the fossa ovalis on your models. But where is this fossa ovalis? Remember, it is separating the right atrium from the left atrium. True story? So where else could I find that fossa ovalis? Inside of that left atrium. And so this model right here has a flap. You open up the left atrium. It's a much bigger atrium, and you got a big portion of the fossa ovalis visible on this side. You guys feel okay with that? By the way, if this, I want you guys to look at your flow charts again. If this is the left atrium, what red blood vessels are going to be associated with the left atrium? What red blood vessels are associated with the left atrium? Look at your flow charts. Pulmonary what though, Alyssa? Pulmonary what? You're totally right. These are pulmonary. Oh, these are pulmonary veins, and yet they're red. Why are they red? Because they're oxygenated. Why are they returning to the left side of the heart? So that the left side of the heart gets all this oxygenated blood into this very large muscular chamber, and it beats and it shoves it all out. Right, we got a different view to share. It is right here. Shoves it all out through the aortic semilunar valve into the aorta. Now the rest of the body has their oxygen. All right, that is the end of my spiel today. Let me go back here, just double check. Base, apex, epimyoendocardium, right atrium, right auricle, right ventricle, left atrium, left auricle, left ventricle, the fossa ovalis that separates the atria, interventricular septum, bicuspid mitral, tricuspid, chordae tendineae, papillary muscles, aortic semilunar valve, pulmonary semilunar valve, aorta, pulmonary trunks, pulmonary veins, vena cava, superior and inferior. Ooh, coronary arteries and coronary sinus. Okay, listen, it's going to be really simple for coronary arteries and coronary sinus. When we get to the blood vessels, you're going to have to learn pretty much all of the blood vessels found on the heart. But for now, It's pretty simple. Just trying to find you guys the proper images for this. This will work really well. So right here, where my arrow is at, is the aorta. It's the ascending aorta. And the ascending aorta has a branch of artery that comes off the right side and it comes off the left side. So this artery you guys see right down here, it's going to circle right around the tricuspid valve. This is the right coronary artery. This is a fantastic view of it. You guys see the aorta right here. And do you see how there is a branch of blood vessel that comes off and a branch of blood vessel that comes off that side? Yes, no, maybe so? So these are going to be the left and right coronary arteries. Great view of it. It's kind of hard to go through. You might have to look up some more internet questions or whatever, pictures. But yeah, that's how we go about finding those. Okay, cool. Now don't forget you have the drag and drop. Let's just do, I don't know, one or two of these together. I don't know. We're kind of out of time and I want to give you guys a breather because it's after 1 o'clock. But I hope you guys can start to see that we have superior vena cava, the ascending aorta, the pulmonary trunk. So all of these things that we discussed. She has a few things on these lists that you did not need to learn. They're not on the objectives list. So should you stress about things like the pericardial sac attachment? For me, yes or no? Nah, of course not, right? Focus on your objectives list. If it's not on your objectives list, pectinate muscles, they're cool. I love them. They're right down here. They're found inside of the auricle, but you don't need to know them. So, boom, we can just kind of set them off to the side. They're not part of what you guys needed to learn. Easy peasy lemon squeezy with practice. Practice? Talk about practice? Now you guys know my reference for that. You talking about practice? Sheep heart stuff. Now the sheep heart can be on the lab practical, but I promise it will look as close to a real model as possible. I'm not going to try to trick you guys. You'll have a big ass heart. It's either going to have the right ventricle, left ventricle, right atrium, left atrium. I'm not going to do anything tricky. If it's super hard to find, I will not test you on it because you don't have a sheep heart in front of you. You're not dissecting it. So it's not fair to me to ask you harder questions. So I will keep it simple. Keep it simple, stupid, right? That's what my dad always told me. Keep it simple, stupid. I'm a nice guy. Yes, ma'am. You're the same group for the rest of the semester. Yep, it's KISS. Keep it simple, stupid. Yep, you're the same group for the rest of the semester. Heather, did you get anybody contacting you from your lab group? Are you with your group? Yes, Alyssa, you got it. Heather, you still there? Yeah, awesome. Thank you, Brianna. Thank you, Bree. Yep, to do today is the blood quiz. Just go on to Blackboard. It should be really easy. And the study guide for endocrine. That's extra credit, so you should be motivated to get it done. Wonderful. What's going to be due Wednesday? The cardio study guide. So you guys got to get on board. Get working on it, okay? You only have 2 days to get this study guide done versus the weekend where you got endocrine. So make sure you guys are working on it. Use the drag and drop, and then use all of these blank pictures that I gave you. Thumbs up Everybody good? Everybody know what they have to do? And then keep working on that homework. Remember, homework you can, I don't even know if I said this. Damn you, Dr. H. I want you to work together on homework. I want you to work as a group. I want groups to have same answers if you all agree on the answer. I want you to use the homework as a study guide. I want you to think about each question and each answer and say, why is it A and not B, C, and D? I want you guys to work together. So don't think you can't work together as a group on homework. You can. I promote it. You cannot work together on an exam, but you can work together on a homework. Is that clear? Do it. Get on Collaborate. Share your screens. Help each other. Don't try to do it yourself. All right, cool. This Friday, I'm going to probably try to get a review session, just a short review session. I'll be working on what time I can. My son has swim in the morning, so it's normally a little afternoon. If you can't make it, it will be recorded, so that's okay. That's okay. All right, I'm out of here. Anybody have any questions? Throw me a text. You're welcome. Hopefully everybody, all righty, all righty, all righty then. Later, y'all.